In the quiet space between wakefulness and sleep, let us drift gently into the world of biochemistry, the study of life's subtle machinery, the chemistry that breathes motion into the stillness of matter. Every heartbeat, every thought, every slow inhale and exhale you take depends upon a delicate choreography of molecules. These molecules are not loud or flamboyant. They move in silence, arranging and rearranging themselves in endless cycles of transformation, sustaining the harmony of life from within. In this soft hour of learning, we will begin to understand the smallest pieces that compose our bodies and how they unite in patterns of astonishing precision and beauty. All living things, from the simplest single-celled organisms to the most complex animals, are built from the same handful of chemical elements: carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. These six, in various combinations, make nearly everything you are: the air you breathe, the food, eat, and the cells that make up your body are all expressions of chemistry. But biochemistry is not merely chemistry. It is chemistry infused with purpose. It is the quiet conversation between molecules that sustains the miracle of being alive. At the foundation of this molecular language are amino acids and proteins. To understand them is to understand the architecture of life itself. But before we arrive there, it helps to pause for a moment and appreciate the world in which they exist. The tiny, bustling environment of the cell. Every cell, no matter how small, is like a city enclosed by walls of delicate membranes. Inside, countless reactions occur simultaneously, each timed and tuned to the needs of the organism. Energy is captured and released, signals are sent and received, materials are transported, and waste is removed, all under the direction of molecules so small that billions of them could fit onto the head of a pin. Imagine this microscopic city under a tranquil light. Floating within its fluid boundaries are long chains of molecules twisting and folding, tiny droplets of fat, coiled strands of genetic code, and countless small fragments of matter, each with a role to play. This is where amino acids and proteins dwell. They are the builders and the workers, the architects and the caretakers of life's molecular structures. Without them, nothing would grow, nothing would heal, and nothing would move. Amino acids are often called the building blocks of life. They are small, simple molecules made of a central carbon atom connected to four groups: a hydrogen atom, a carboxyl group, an amino group, and a variable side chain that defines their identity. Though this arrangement may sound technical, there is a quiet elegance to it, a balance between simplicity and potential. Out of just 20 types of amino acids, nature has composed every protein found in every living organism on Earth. From the keratin that shapes your hair and nails, to the enzymes that digest your food, to the antibodies that guard you against disease. All of them are built from these 20 molecular letters, combined and recombined in endless variation. Biochemistry, at its heart, is the study of how these letters come together to write the language of life. It is a language that does not use words or sounds, but shapes and charges and affinities. When amino acids join, they form proteins, and these proteins fold into forms that determine what they can do. Some fold into strong fibers that lend support and structure. Others curl into precise globes that perform delicate chemical reactions. Every shape tells a story, and every story sustains life. To imagine this at work, think of a river gently flowing through a forest. Along its banks, plants grow, animals drink, and unseen beneath the surface, countless organisms thrive. The river never stops moving, and yet its rhythm is calm and constant. Biochemical processes are much the same. Body, proteins, and amino acids are constantly forming, folding, breaking, and reforming. Molecules meet and part again in cycles of creation and renewal, maintaining balance with every passing second. The word biochemistry might sound heavy, but it really means something very gentle: the chemistry of life. It explores how simple atoms come together to create complexity, how order arises naturally from apparent chaos. It shows us that every cell is a small miracle of organization, where thousands of reactions happen in harmony, never colliding or interrupting, but weaving together like threads in a tapestry. And at the heart of that tapestry lies the story of proteins, woven from chains of amino acids linked by bonds so precise that even a single change can alter the entire pattern. These bonds form through a process known as dehydration synthesis. When two amino acids meet, they link together by releasing a single molecule of water. One molecule's carboxyl group offers a carbon, the other's amino group offers a nitrogen, and together they form what is called a peptide bond. It is such a small act, one tiny drop of water set free, yet through millions of these connections. It is humbling to think that the body, with all its organs and sensations, its memories and movements, is made from a pattern that begins with such simplicity. Proteins made from these linked amino acids serve as the functional machinery of life. They are not passive structures. They move, bend, and interact. Some act as enzymes, accelerating the countless reactions that sustain metabolism. Others form filaments and tissues, giving strength and shape to skin, muscles, and bone. Still others carry messages between cells or transport oxygen through the bloodstream. If DNA is the blueprint of life, proteins are the builders that turn those instructions into reality. In a sense, the study of proteins is also the study of form and beauty. When scientists observe them under powerful microscopes, what they see are intricate spirals, coils, and sheets folded upon themselves in perfect geometry. Each fold is dictated by subtle forces, attractions between charges, the desire of water to cling or to avoid, the balance of tension and release. The result is a structure so exact that even a small disturbance can change its entire purpose. And yet, despite this fragility, life endures. Proteins fold correctly billions of times a day within your body, a testament to nature's precision and grace. As we begin this journey, it is helpful to remember that biochemistry is not separate from us. It is not an external science that we observe from afar. It is happening within you, in every cell, at every moment. When you breathe, proteins guide oxygen into your blood. When you digest, enzymes break food into nutrients. When you rest, amino acids repair and rebuild your muscles. Even as you listen now, as your eyes grow heavy and your frame, thoughts drift toward sleep, biochemical reactions continue their quiet work, keeping the rhythm of life steady and sure. In the chapters to come, we will explore this molecular world more deeply. We will meet the amino acids one by one, understand how they join, how they fold, and how their collective behavior gives rise to everything we recognize as living. We will see how proteins form elegant architectures inside the cell, how they communicate, and how they adapt to the environment around them. But for now, as this first part draws to a close, allow yourself to rest in the knowledge that within you exists a vast and silent symphony of chemistry. Each note played by an amino acid, each melody shaped by a protein's gentle curve. Together, they sustain the harmony of life, a harmony so intricate and yet so effortless that it has continued unbroken since life first appeared on Earth, billions of years ago. So let your breathing slow. Imagine the countless molecules within you, moving in graceful balance. The amino acids drifting, connecting, forming bonds, and folding into the beautiful structures that make you who you are. And as you drift deeper into calm, remember that this is not only science, it is also a story, a quiet tale of creation written in the language of chemistry, whispered endlessly inside every living cell. 2. The essence of amino acids. In the stillness of the mind, let us wander deeper into the heart of biochemistry, to the quiet, intricate simplicity of amino acids, the true origin of life's endless variety. They are the smallest architects of all living structure, the elemental notes in life's molecular symphony. Though invisible to the eye, they shape everything that breathes, grows, and remembers. Every strand of hair, every muscle fiber, every enzyme quietly turning within you, all of it begins with amino acids. To understand them is to glimpse the elegance of life reduced to its most graceful form. An amino acid may seem simple on paper, but in its simplicity lies perfection. Each molecule contains a single carbon atom at its center, known as the alpha carbon, like the trunk of a small tree from which all other parts extend. From this central point stretch 4 branches: one hydrogen atom, one amino group, one carboxyl group, and one variable side chain, the R group, as scientists call it. 3 of these branches are constant, present in every amino acid that has ever existed. But the 4th, the side chain, is what gives each amino acid its distinct identity, its particular flavor of behavior. There are 20 of these amino acids used universally by living things, 20 small yet distinct personalities. Each one contributes something different to the character of the protein it joins. Some are gentle and shy, avoiding water and folding themselves deep inside a protein's heart. Others are bold and charged, drawn to the outer surface where they can interact with the watery world of the cell. Some are flexible, allowing the protein to bend and move. Others are rigid, giving structure and stability. Together, they create an infinite palette of form and function. The side chains, those subtle variations of shape and chemistry, are what make amino acids so extraordinary. Some, like glycine, carry only the simplest side group, a single hydrogen atom, making them small, nimble, and adaptable. Others, such as tryptophan, are complex, bearing large rings of carbon and nitrogen that lend weight and richness to the structure of proteins. A few carry electric charges, like lysine and glutamic acid, allowing them to form tiny attractions and repulsions that help proteins fold precisely into shape. Some prefer water, others shy away from it. Some are acidic, others basic, and in these differences lies the quiet genius of biological design. Imagine, for a moment, a painter's palette with only 20 colors. It might seem limited at first, yet from those few shades, the painter can create endless landscapes and portraits. Every subtle hue of light and shadow. Nature does much the same with amino acids. With just 20 molecular colors, it paints the entire spectrum of life: the silk of a spider's web, the strength of bone, the pulse of enzymes driving the chemistry of thought. Amino acids do not exist in isolation for long. The amino group of one meets the carboxyl group of another, and between them, a bond is formed. It is called a peptide bond, small, precise, and incredibly strong for its size. This joining releases a molecule of water, a quiet reminder that in creation, something is always given up. This process continues, one amino acid after another joining the growing chain, until a long thread is formed. That thread, though simple at first, will one day twist and fold into the shape of a living protein. Each amino acid in this chain knows its place. Its side chain determines how it interacts with its neighbors, whether it clings to them tightly, repels them, or curls away in a different direction. As these molecular forces balance and adjust, the chain begins to fold upon itself. The result is not chaos, but order, a shape so precise that it repeats perfectly every time, as if the molecule remembers how to fold. And in truth, it does. The sequence of amino acids holds all the information needed for that folding, written into the chain like a secret code. The sequence of amino acids, known as the primary structure of a protein, is dictated by the instructions stored in DNA. Within the nucleus of each cell, long strands of genetic material record the order in which these building blocks must assemble. through a process called translation. This information is read and transformed into a chain of amino acids by tiny cellular machines known as ribosomes. The ribosome moves along the code like a reader tracing words on a page, adding one amino acid at a time, following the instructions with patient precision. When the chain is complete, it detaches, curls, and begins to find its destined shape. It is astonishing that such tiny entities could possess so much information. A single protein may contain hundreds of amino acids, each one placed exactly where it must be, no more and no less. If even one amino acid is changed, the result can be profound. The difference between a healthy form of hemoglobin and the one that causes sickle cell anemia, for instance, comes down to a single substitution, one amino acid replaced by another in a chain of more than 100. It is a delicate reminder that in life's design, the smallest variations can ripple outward into vast consequences. Beyond their role in building proteins, amino acids have lives of their own within the body. Some act as precursors to hormones, molecules that carry signals between cells. Others form neurotransmitters, helping messages pass along the sparse, intricate pathways of the brain. The amino acid tryptophan, for instance, can be converted into serotonin, a molecule that influences mood and calmness. Another, tyrosine, gives rise to dopamine and adrenaline chemicals that shape alertness, focus, and motion. Even the chemistry of feeling itself, it seems, begins with these modest molecules. In the body, not all amino acids are created equally. Some are known as essential, meaning they must be obtained through the diet, because the body cannot produce them on its own. These include lysine, methionine, and valine, among others. They arrive from the food we eat from grains, beans, vegetables, and animal proteins entering the bloodstream and finding their way to cells in need of repair or growth. Others, the non-essential amino acids, can be synthesized by the body itself, built from simpler compounds already present. Together, these two groups maintain the constant renewal of tissues, the repair of muscles, and the production of enzymes that keep life in balance. In this way, amino acids are both the substance and the story of nourishment. Each meal you take, each breath, contributes in some way to replenishing them. Within hours, the proteins from food are broken down into amino acids, which then circulate quietly, waiting to be used. A muscle in need of repair may draw upon them. The liver, ever busy, may rearrange them to create enzymes for detoxification. Even during sleep, your body's silent factories are at work, recycling and rebuilding, ensuring that each cell wakes renewed. If we could listen to the whispers of chemistry, the language of amino acids would sound like a song of balance and cooperation. Every molecule knows how to find its place, guided by the invisible rules of attraction and repulsion. Hydrogen bonds form like gentle handshakes, holding structures in place. Hydrophobic forces draw some molecules inward, away from water, while others are drawn outward, eager to touch, get it. Ionic interactions, tiny electrical pulls, adjust the positions of side chains, like dancers moving in rhythm. Together, these interactions shape the elegant geometry of proteins, the spirals, sheets, and loops that define the architecture of life. As your mind settles, imagine the interior of a single cell, a quiet ocean filled with movement. In that space, amino acids drift freely until called upon. Some gather to form enzymes, others to build fibers of muscle or collagen, others still to serve as messengers. They are never idle. Their world is a place of constant transformation, where structure and purpose emerge from fluid motion. Amino acids remind us that creation often begins from simplicity. They show that even the smallest units can hold infinite potential. They are the bridge between the lifeless chemistry of elements and the living wonder of biology. As this, chapter draws to its gentle close. Picture the 20 amino acids drifting like stars in a soft molecular sky. Some glow with polar attraction, some shimmer with neutrality, others sparkle with charge and movement. They wait patiently, ready to join hands, to link and fold and become something greater than themselves. In the next part of our journey, we will watch as they come together through bonds and structures, forming the first delicate threads of proteins, the quiet weavers of all living form. 3. The architecture of life's building blocks. In the unbroken stillness of the microscopic world, there exists a harmony that shapes all living things. A quiet, invisible architecture, built not from bricks or stone, but from strings of amino acids folded into intricate and beautiful forms. These structures, called proteins, are the true builders of life. Every heartbeat, every movement of muscle, every thought in the brain depends upon them. They are the silent artisans, working within each cell, crafting, repairing, and maintaining the entire living body, from the smallest bacterium to the vast complexity of a human being. To see proteins as they truly are, one must imagine a world beyond sight, a world where the tiniest vibrations carry meaning, and shape is everything. When amino acids link together, they form chains known as polypeptides. These chains may seem simple at first glance, just sequences of small molecules bound end to end, but within each sequence lies a hidden blueprint, a set of instructions that tells the molecule how to fold, twist, and coil into a specific form. The form determines the function, and the function, in turn, sustains life. The order of amino acids along a polypeptide chain is known as the primary structure of a protein. It may be compared to the letters in a sentence, meaningless on their own, but filled with purpose when arranged in the right order. Even a single change, one letter out of place, can alter the entire message. So too can one misplaced amino acid transform a protein's behavior completely. The elegance of this molecular language lies in its precision. Every living cell must know how to assemble these sequences exactly, guided by the code of its DNA, so that the resulting proteins can perform their tasks flawlessly. Once the sequence is complete, the protein begins to fold upon itself, seeking the most stable form it can achieve. This folding happens naturally, without conscious direction, guided by the subtle forces between atoms. Some amino acids are drawn to water, while others shy away from it. Some carry small electric charges that attract or repel their neighbors. These quiet interactions pull the chain into bends and loops, forming the first hints of structure patterns that repeat, like gentle ripples in water. These early shapes are called secondary structures, and two of them appear most often: the alpha helix and the beta sheet. An alpha helix resembles a spiral, staircase, each step supported by hydrogen bonds that keep the structure steady and compact. Beta sheets, by contrast, look like pleated ribbons, flat and extended yet strong. Within a single protein, these patterns may appear and reappear, twisting and combining in endless variations. The beauty of this process is its spontaneity. The protein needs no external help to find its shape. The information for folding is written into its sequence, as though the molecule remembers what it is meant to be. When these smaller patterns combine, the protein takes on its full, three-dimensional shape, what scientists call its tertiary structure. Here, the chain folds into a compact form, with some regions buried deep inside and others exposed to the watery environment of the cell. The result is not random but purposeful. Every fold, every curve, every pocket has meaning. A crevice might become the active site of an enzyme, the spot where a reaction takes place, docking site for another molecule. These forms, though invisible to the naked eye, are as real and intricate as the architecture of a cathedral. Elegant, functional, and built to endure. Sometimes, several folded chains join together to form an even larger complex, known as the quaternary structure. Hemoglobin, the protein that carries oxygen in the blood, is one such masterpiece. It is composed of 4 subunits, each holding an iron atom at its center. Together, these subunits work in perfect unison, picking up oxygen in the lungs and releasing it where it is needed. The arrangement is cooperative and rhythmic, like a small orchestra playing in time. If one subunit changes shape slightly, the others follow, ensuring that the protein's performance remains smooth and balanced. The variety of protein structures found in nature is almost infinite. Some are long and fibrous, built for strength and durability. These include collagen, which gives structure to skin and tendons, and keratin, which forms hair, nails, and feathers. Others are compact and globular, their folded surfaces ready to interact with other molecules. Enzymes, antibodies, and hormones all belong to this family. Some proteins serve as channels in cell membranes, allowing ions to pass through like travelers moving along a narrow road. Others act as messengers, translating signals between cells or triggering reactions within them. What unites all these forms is the delicate relationship between structure and function. If the shape of a protein changes even slightly, its behavior can change entirely. Heat, acidity, or certain chemicals can disrupt the bonds that hold a protein together, causing it to unfold in a process called denaturation. This unfolding is often irreversible, and the protein loses its ability to function. You can see a simple example of this in the kitchen. When an egg cooks, the clear liquid turns solid white as the proteins in it denature and refold into new, tangled shapes. The chemistry of life is fragile, and its beauty lies in how carefully balanced it must remain. Inside your body, however, this balance is maintained with extraordinary precision. Molecular chaperones, special proteins themselves, guide other proteins into the correct shapes, ensuring that they do not misfold or clump together. They are the silent caretakers of cellular order, keeping chaos at bay in the microscopic world. Without them, the cell's interior would become crowded with tangled molecules, unable to perform their roles. This careful guidance mirrors the way nature itself tends toward harmony, even at the smallest scale. Over billions of years, small changes in DNA sequences have altered the amino acid patterns in proteins, producing new shapes and functions. From a single ancestral protein, thousands of variations can emerge, each adapted to a particular need: the silk of a spider, the venom of a snake, the enzymes that break down sugar in your blood. Yet despite this diversity, all proteins share the same underlying principles: the same 20 amino acids, the same rules of bonding and folding. It is a universal architecture, repeated endlessly, refined with time. To imagine this on a more personal level, think of your own body as a living gallery of molecular design. The collagen in your skin keeps it supple. The actin and myosin in your muscles allow them to contract. The hemoglobin in your blood carries oxygen with every breath. Even the smallest motions, the flicker of an eyelid, the soft beating of your heart, depend upon proteins, sliding past one another, folding, binding, and releasing in perfect coordination. You are, in a very literal sense, a moving sculpture shaped by proteins. Beyond their physical roles, proteins also embody the principle of transformation. They are in constant motion, never static, shifting subtly to accommodate other molecules. When an enzyme meets its substrate, the molecule it acts upon, both change shape slightly to fit together, like two dancers meeting in step. This adaptability, known as induced fit, allows reactions to occur quickly and efficiently. It is as though the protein breathes in response to its environment, sensing what is needed and adjusting accordingly. This quiet adaptability reveals something profound about life itself. The molecules that sustain us are not rigid mechanisms, but fluid, living forms. They move and respond, balancing between order and flexibility. Every protein, in its folding and unfolding, embodies this delicate dance between structure and motion. And it is this dance that makes life possible, the constant interplay of shape, energy, and purpose. As we drift toward the end of this reflection, imagine the inside of a single cell once again. There, countless proteins float and twist in the cytoplasm, each finding its place, each performing its task. Some assemble into filaments, others catalyze reactions, others still carry messages from one compartment to another. It is a city built not of steel or stone, but of motion and connection. The architecture of life is soft, fluid, and alive, always forming, always reforming, in a rhythm that mirrors the pulse of existence itself. When you next breathe deeply and feel the steady strength of your body, know that this strength is not made of solid walls, but of flowing molecules bound together by invisible forces. The architecture of life's building blocks is both simple and miraculous, a reminder that beauty, at every level, begins in order, balance, and quiet motion. 4. Peptide bonds and the music of connection. In the tranquil depths of biochemistry, every connection tells a story. Bonds are not mere lines between atoms. They are threads that hold life together, links in the vast molecular web that keeps the body whole and harmonious. Among the countless types of bonds that form and break in living systems, few are as quietly significant as the peptide bond, the subtle tie that joins one amino acid to another, creating the long, delicate chains we know as proteins. To understand this bond is to listen to the soft music of connection itself, a molecular rhythm that began billions of years ago and continues, unbroken, within every living cell. When two amino acids approach each other, they do not collide at random. They meet through precise chemical alignment, like dancers finding their steps in a slow, patient waltz. One amino acid carries a carboxyl group at one end, a carbon atom double bonded to an oxygen and connected to another oxygen through a hydroxyl group. The other amino acid bears an amino group containing nitrogen and hydrogen atoms. As these two approach, the carboxyl carbon of one molecule aligns perfectly with the amino nitrogen of the other. Then, in a moment of molecular grace, a single molecule of water is released, a hydrogen from the amino group, an oxygen and hydrogen from the carboxyl group, and in their place, a new connection forms. This connection, this bridge of shared electrons, is the peptide bond. It is a simple reaction in essence, called a dehydration synthesis, because it builds by removing water. But in its simplicity lies elegance and permanence. The resulting bond is remarkably stable, resisting spontaneous breakdown even under conditions that would disrupt many other molecular connections. This durability is vital, for the chains that form through peptide bonding must remain intact through the turbulence of cellular life, through heating, cooling, folding, and unfolding, through all the gentle chaos that occurs within the cell's fluid interior. As each new peptide bond forms, the chain grows longer, one amino acid at a time. The sequence unfolds like a sentence being written letter by letter, each addition guided by the ribosome, a molecular machine that reads the genetic code. Inside this tiny factory, amino acids are ferried into place by transfer molecules known as tRNAs. Each tRNA carries its specific amino acid, matching it to the correct sequence on a strand of messenger RNA. The ribosome moves steadily along this message, joining one amino acid to the next, forming peptide bonds with rhythmic precision. In this way, the language of DNA becomes the language of proteins, and chemistry becomes life's poetry. To imagine this process, picture a loom weaving an endless thread. Each amino acid is a new thread added to the growing tapestry. The peptide bond acts as the weaver's knot, invisible but indispensable, holding the pattern together. The finished chain, the polypeptide, may be hundreds, even thousands, of amino acids long, and yet, Though each peptide bond is identical in form, the sequence of amino acids gives every chain a unique identity, much as a melody takes shape from notes arranged in a particular order. The strength and stability of peptide bonds give proteins their remarkable resilience, but these bonds also impart flexibility. The atoms on either side of the bond can rotate around specific axes, allowing the entire chain to bend and fold. This subtle freedom of movement is what enables proteins to form their intricate shapes, curling into helices, pleated sheets, and complex three-dimensional structures. It is the balance between rigidity and flexibility that gives proteins their lifelike quality, firm enough to hold a shape, supple enough to move and respond. If we could shrink down to the molecular scale, we would see these bonds shimmering with energy, connecting one small molecule to another in endless patterns. In a single cell, there are billions of peptide bonds, forming and reforming as new proteins are made and old ones are broken down. The cell never stops building. It is a place of constant creation, where each connection contributes to the quiet hum of life. This ceaseless activity is not random. It is directed by the genetic blueprint written within the DNA. The sequence of bases, adenine, thymine, cytosine, and guanine, determines the sequence of amino acids and therefore the order of peptide bonds that will form. The precision of this process is astonishing. A single misplaced bond could alter the folding of a protein, changing its structure and, by extension, its function. Life depends on the accuracy of these bonds, and nature has evolved elaborate systems to check and correct mistakes. The energy required to form peptide bonds is not drawn from chance but supplied by specialized molecules like ATP. In the ribosome, chemical energy is carefully converted into the mechanical work of building proteins. The peptide bond, once formed, stores a small portion of this energy, ready to be released later when the protein is eventually degraded. It is part of life's elegant economy. Nothing wasted, everything reused. The beauty of the peptide bond is that it links not just atoms, but meaning. Each bond represents continuity, one molecule giving a part of itself to another, both changed by the connection. The released water molecule, drifting away into the cellular fluid, is a gentle reminder that creation often involves letting go, and in that small act of release, something greater is born. When many peptide bonds come together, the chain they form begins to resemble a living entity. It folds upon itself, guided by forces within and around it. Hydrogen bonds form between nearby sections, stabilizing delicate helices and sheets. Interactions among side chains, hydrophobic, polar, acidic, and basic, shape the chain further, drawing it into loops and folds until the final form emerges. The peptide bonds themselves, though hidden deep within the structure, remain the backbone, the unseen framework upon which the entire protein rests. In the human body, peptide bonds are everywhere. They form the muscle fibers that contract with every heartbeat, the enzymes that digest food, the hormones that carry messages between organs. Even the collagen that supports skin and bone is made of long, coiled chains held together by countless peptide bonds. These connections endure through years, sometimes decades, before being replaced. Every moment of life depends on their quiet strength. The stability of peptide bonds also has another profound effect. It allows the body to store information in physical form. A protein's shape, determined by its sequence of peptide bonds, encodes instructions for how it should act, where it should go, what it should do. In this way, biology achieves a form of memory, not in words or symbols, but in the geometry of molecules. When one protein recognizes another, when an enzyme fits its substrate, or a receptor binds a hormone, it is this molecular memory that guides the interaction. In the broader rhythm of nature, peptide bonds have been forming for billions of years. They appeared in the earliest oceans, When the first amino acids encountered one another and found ways to link, from those primordial chains came the first functional proteins, and from those proteins came cells, tissues, organisms, and the vast web of life we know today. Each bond, then, is not just chemistry. It is a continuation of that ancient melody, the same song that began when Earth itself was young. As your thoughts begin to slow, imagine this molecular music playing softly within you. Each peptide bond is a note in the symphony, connecting one sound to the next, forming the endless score of your existence. The proteins within your cells hum quietly as they build, repair, and renew, their movements in perfect time. The water released with each bond evaporates like a sigh, and the chain grows longer, stronger, and more beautiful with every step. The music of connection never ends. It flows through you in every breath, every pulse, every cell. The peptide bond is its rhythm, steady and enduring, the heartbeat of life, written not in sound but in structure. And as you drift deeper into rest, know that this music continues within you, soft and eternal, binding molecule to molecule, self to self, in the serene harmony of existence. 5. The 4 levels of protein structure. Within the delicate architecture of life, proteins stand as the most remarkable of all biological creations. They are the quiet instruments of existence, crafted from threads of amino acids that fold and twist into forms of infinite complexity. Every breath, every heartbeat, every thought is made possible by their silent labor. Yet beneath this vast diversity, there is a beautiful order, a hierarchy of structure that governs how a simple chain of molecules becomes a living, functional entity. Scientists describe this as the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. These levels are not separate, but continuous, one flowing gently into the next, like ripples expanding across still water. The story begins at the most fundamental level, the primary structure. Here, we find the protein in its simplest form, a linear chain of amino acids connected by peptide bonds. This sequence, though seemingly unassuming, carries the entire blueprint of the protein's identity. Each amino acid is a letter in a sentence, arranged with absolute precision. The order in which these letters appear determines everything that follows: how the chain will fold, how it will move, and what role it will play within the cell. A single substitution, even the exchange of one amino acid for another, can transform the entire meaning of the sequence. In the body, this truth is both powerful and fragile. The condition known as sickle cell anemia, for instance, arises from a single change, one amino acid replaced by another, altering the structure of hemoglobin and changing the shape of red blood cells. Such is the influence of the primary structure. The smallest detail can echo throughout the entire organism. Once the chain is complete, nature begins to fold it into form. The next step in this graceful process is the secondary structure, the first stage of organization within the molecule's long, flowing body. Here, the amino acid chain bends and loops upon itself, guided by hydrogen bonds that form between atoms of the backbone. These bonds do not involve the side chains, but rather the repeating pattern of nitrogen, hydrogen, and oxygen that make up the chain itself. Through their quiet attractions, they create repeating motifs that stabilize the structure. 2 patterns dominate this level: the alpha helix and the beta pleated sheet. The alpha helix is a delicate spiral, coiled like a ribbon or a spring. Each turn of the helix is held in place by hydrogen bonds, giving it strength and elasticity. In contrast, the beta sheet lies flat and extended, formed by parallel or anti-parallel strands connected through rows of hydrogen bonds. The result resembles a softly folded fabric, flexible yet firm. These recurring patterns are the first hints of order in what was once a simple chain. They appear spontaneously, as though the molecule remembers how it is meant to shape itself. Within the confines of the cell, countless proteins form these helices and sheets, each one adding stability and definition to the growing structure. But nature does not stop there. The secondary structures begin to interact, drawing close through gentle forces that pull and shape the molecule into a more complex form. This third stage, the tertiary structure, is where the protein takes on its true three-dimensional shape. Here, the side chains of the amino acids, those variable groups that give each one its unique personality, come into play. Some are drawn to water, some repelled by it, some carry charges, positive or negative, that attract or repel one another. Others form bridges of sulfur, known as disulfide bonds, which lock distant parts of the molecule together. All of these forces act in harmony, guiding the folding process with astonishing precision. The tertiary structure is the level at which form and function meet. It is here that the protein's surface takes shape, its grooves, pockets, and folds, each perfectly suited to the tasks it must perform. Enzymes, for example, have active sites formed at this level, small indentations where specific molecules can bind and reactions can take place. Antibodies fold in ways that allow them to recognize foreign invaders with exquisite accuracy. Structural proteins, like those in skin and hair, align their folds into strong, durable patterns. Every curve, every twist, every hidden corner of the tertiary structure has a purpose, shaped by the chemical properties of its parts. The folding of a protein into its tertiary form is a process of quiet artistry. It happens spontaneously in many cases. Within the cell, special helper proteins, called chaperones, oversee the process, ensuring that the folding occurs correctly. These chaperones do not form part of the finished structure. Instead, they act as guides, preventing the newly formed chain from tangling or misfolding. The cell is a crowded place, filled with countless molecules jostling for space. Without these careful attendants, many proteins would lose their way, folding incorrectly, and becoming useless or even harmful. With the chaperone's help, the molecule finds its intended shape, stable and ready for its role. For some proteins, this is the end of the journey. They function perfectly as a single folded chain, but others must go further, joining with additional chains to form something greater than themselves. This final stage is the quaternary structure, where multiple polypeptides come together into a functional whole. Each individual chain, now called a subunit, interacts with others through non-covalent forces or disulfide bridges, forming a larger, cooperative assembly. The quaternary structure is the level of teamwork and harmony, where many parts combine to perform a single, unified function. One of the most familiar examples of this level is hemoglobin, the oxygen-carrying protein in red blood cells. It is composed of 4 subunits, 2 alpha chains, and 2 beta chains, each holding a heme group with an iron atom at its center. When one subunit binds to oxygen, the others change shape slightly, making it easier for them to bind as well. This coordinated behavior allows hemoglobin to pick up oxygen efficiently in the lungs and release it gently in the tissues. The beauty of the quaternary structure lies in this cooperation, the way each part responds to the others. The 4 levels of protein structure are not merely scientific categories. They are the unfolding of a natural design. From the linear simplicity of the primary sequence to the elaborate assemblies of the quaternary form, each stage builds upon the last in quiet perfection. The journey from one level to another mirrors the progression of life itself, from simplicity to complexity, from potential to realization. Within every living cell, these transformations occur unceasingly, creating the endless variety of forms that make up the world around us. In truth, the beauty of protein structure lies in its unity. Every level, from the sequence of amino acids to the cooperative dance of subunits, is part of a single continuum. No level can exist without the others. The stability of the peptide bonds holds the chain together. The hydrogen bonds, of the secondary structure give it texture. The side chain interactions of the tertiary form shape its identity, and the associations of the quaternary level bring purpose and harmony. Together, they create a structure that is alive, not in the human sense of the word, but in the way that it responds, adapts, and endures. As you rest and imagine this microscopic world, picture a protein folding slowly in the gentle fluid of a cell. First, a simple line, then a curl, a twist, a fold, and finally, a complete form. Imagine the forces that hold it together, the tiny attractions between atoms, the quiet tension of bonds, the subtle interplay of water and charge. Each protein completes its folding in silence, then goes on to serve its role, perhaps for minutes, perhaps for years, and when its time ends, it unravels once more, its amino acids set free to begin the cycle again. Life is made of these silent transformations, one structure giving rise to another, endlessly renewed. The 4 levels of protein structure are the stages of this eternal rhythm, the progression from raw material to living design. They remind us that order can arise from simplicity, and that in the smallest corners of existence, beauty is not imposed but emerges naturally. The architecture of life, built molecule by molecule, continues its patient work within you now, as it has since the beginning of time. 6. The language of folding. In the quiet spaces of the cell, where molecules drift like silent travelers through a sea of life, there unfolds one of the most elegant acts in all of biology, the folding of proteins. It is an invisible choreography, a dance so intricate and precise that it happens billions of times every moment across all living things. Every cell, every organism depends on this quiet process. Protein folding is nature's secret language, a language written in the subtle grammar of chemistry, translated by the forces of attraction and repulsion, and spoken in the soft movements of matter becoming form. When a chain of amino acids first emerges from the ribosome, it resembles a slender thread, fragile and unassuming. At this early stage, it has no defined shape, no function, no identity. It is potential waiting to be realized. Yet within this simple sequence lies a perfect memory. The precise order of amino acids determines exactly how the chain will fold into its final shape. The information for folding is not stored anywhere else. It is written into the sequence itself, as though the molecule remembers what it is meant to become. The process begins the moment the chain leaves the ribosome and touches the watery environment of the cell. From that instant, invisible forces begin to act, guiding it toward the most stable arrangement of its parts. Folding is driven by the delicate balance of energy. The chain seeks a shape in which its internal energy is minimized, a state of calm, where every force finds equilibrium. The hydrophobic side chains, which avoid water, move inward, curling away from the surrounding fluid. The hydrophilic, water-loving residues turn outward, reaching toward the solvent. Charged side chains form pairs of attraction, while uncharged ones drift quietly between them. Hydrogen bonds spring into place, linking distant sections of the chain. The result is a slow, deliberate transition from chaos to order, as the molecule folds in upon itself like a ribbon carried by the wind. Imagine a world at this scale. You would see the chain twisting and turning, parts of it collapsing inward, others unfolding and trying again. The motion is neither random nor mechanical. It is graceful and fluid, a dance shaped by chemistry's quiet laws. In this molecular ballet, every move has purpose. The chain folds not by trial and error alone, but by a series of guided steps, intermediate shapes that lead naturally toward the final form. These fleeting configurations, called folding intermediates, are like the half-formed gestures of a dancer finding balance before the final pose. Folding is astonishingly efficient. In nature, a protein can find its correct shape within milliseconds or seconds, even though the number of possible configurations is astronomically large. If a molecule were to explore every possible arrangement one by one, the process would take longer than the age of the universe. Yet somehow, in the gentle currents of the cell, the correct form emerges almost instantly. This remarkable speed suggests that folding follows specific pathways, like a traveler moving downhill through a landscape of energy, always seeking the lowest valley. The energy landscape of protein folding is often imagined as a funnel wide at the top, where many shapes are possible, and narrow at the bottom, where the stable, functional form awaits. The molecule slides down this funnel, guided by forces that pull it toward its destiny. The crowded interior of a cell is filled with other molecules, and collisions are inevitable. A newly formed protein, if it folds incorrectly or clumps with its neighbors, can lose its function entirely. In severe cases, such misfolding can cause harm. Certain diseases, such as Alzheimer's and Parkinson's, are linked to proteins that have misfolded and formed aggregates, tangled clusters that disrupt cellular function. Yet these events are the exception, not the rule. In the vast majority of cases, folding proceeds flawlessly, safeguarded by special helper molecules called chaperones. Molecular chaperones act like quiet guardians of order. They do not dictate the protein's final shape, but they ensure that folding occurs safely, shielding vulnerable chains from interference. Some chaperones form small chambers, protective enclosures where the protein can fold in peace, away from the bustling traffic of the cytoplasm. Others bind temporarily to unstable regions of the chain, preventing them from sticking to the wrong partners. When the folding is complete, the chaperones gently release their charge, allowing the newly formed protein to drift away and begin its work. It is an act of precision and patience, repeated endlessly within every living cell. As the protein folds into its final structure, layers of order appear: helices coiling, sheets aligning, loops connecting one region to another. Each bond, each fold, contributes to the whole. The forces at work are subtle yet powerful. Hydrogen bonds hold secondary structures in place. Hydrophobic interactions stabilize the core. Ionic bonds connect distant parts, and van der Waals forces fine-tune the shape. The final product is a molecule perfectly shaped for its purpose. An enzyme with a pocket that fits its substrate, a receptor ready to recognize a signal, or a structural protein strong enough to support the weight of life. The folded protein, once complete, is not rigid. It breathes and moves, shifting its shape slightly as it performs its functions. Some regions are flexible, designed to change in response to their environment. When a molecule binds to a protein as a substrate to an enzyme or a hormone to its receptor, the protein adjusts its conformation subtly to fit, like a hand closing around an object. This adaptability is a hallmark of life's design. The molecule is both stable and dynamic, a structure that can endure yet respond, maintaining balance in an ever-changing world. The elegance of folding also extends beyond the individual protein. In the crowded interior of the cell, thousands of proteins fold and unfold in unison, their shapes forming a living architecture. Some join to form filaments and scaffolds that give the cell its structure. Others assemble into machines that carry out complex tasks, such as replicating DNA or producing energy. Each of these assemblies depends on the precise folding of its parts. A single error in one protein structure could compromise an entire system, reminding us how delicate and interconnected life truly is. There is something deeply poetic about this process. The folding of a protein is not driven by consciousness or intention, yet it produces outcomes of astonishing purpose. It is as if nature itself understands the language of harmony, every hydrogen bond, every twist in the chain. Through this language, the cell speaks silently, building, repairing, and renewing itself without pause. It is a song of balance, sung not in sound, but in motion. As you picture this quiet folding, imagine the smooth flow of time within the cell. The chaperones release it, and it takes its place among the others, fitting perfectly into the greater whole. The process happens unseen, yet it is as constant as the rhythm of your breathing. Even now, as your body rests, thousands of new proteins are forming within you, each one folding itself with unerring grace. Life depends on this invisible language, the language of folding. It is what transforms raw chemistry into living design, what turns the randomness of molecules into the order of thought, growth, and renewal. It is a reminder that within the smallest details of existence, there is intelligence beyond measure, not the intelligence of minds, but of matter, knowing how to find balance, how to become what it must be. As you let this image settle in your thoughts, they are the living threads of your being, each one shaped by the forces of the universe, each one performing its silent duty. The language of folding continues within you, wordless and ceaseless, a conversation that began when life itself began, and one that will continue as long as life endures. 7. Enzymes, the quiet catalysts of life. Deep within the stillness of every living cell, beneath layers of structure and in movement, it is a labor without rest, one that keeps every process of life in motion, the transformation of food into energy, the repair of tissues, the transmission of thought, the slow, patient renewal of every molecule. This unseen work is carried out by enzymes, the silent catalysts of existence. Without them, the chemistry of life would halt completely, for enzymes are what make the improbable possible, turning sluggish reactions into swift, purposeful flows of energy and transformation. They are, in the truest sense, the guardians of life's rhythm, unseen, tireless, and precise. To understand enzymes is to understand the power of subtlety. In the chemical world, reactions require energy to begin, a small push that allows bonds to break and new ones to form. This energy is called the activation energy, a kind of barrier that prevents chaos by ensuring that molecules do not react randomly. But within living organisms, time cannot wait for slow chemistry. A single reaction that might take years to occur on its own must happen in moments for life to continue. Enzymes lower this barrier, making it easier for reactions to take place. They do not force the process, they guide it, like a gentle hand turning a key in a lock. Each enzyme is a protein shaped precisely for the reaction it controls. Its surface is a landscape of curves and hollows, ridges and valleys, all arranged to recognize a specific molecule, its substrate. When the substrate drifts close, it fits into a region called the active site, a small pocket, perfectly matched in shape and charge. The two come together as naturally as a seed settling into soil. For a brief moment, they form a complex enzyme and substrate joined in silent cooperation. Then, almost effortlessly, the reaction occurs. Bonds rearrange, atoms shift, and a new product is formed. When the transformation is complete, the enzyme releases its product and returns to its original state, ready to begin again. This precision is what makes enzymes so extraordinary. Each one is designed for a single purpose, recognizing only the molecule it was meant to act upon. The enzyme sucrase, for instance, breaks down sucrose common table sugar into glucose and fructose. The enzyme lipase targets fats, cutting them into smaller components that can be used for energy. DNA polymerase builds the long chains of genetic material that store the instructions of life, while RNA polymerase transcribes those instructions into a language the cell can use. Each enzyme knows its role, and each performs it with quiet perfection, millions of times over. The relationship between an enzyme and its substrate is often described through the lock and key model. In this image, the enzyme's active site is the lock, and the substrate is the key. Only the correct shape will fit. But nature, as always, allows for nuance. Enzymes are not rigid structures. They are flexible and alive, capable of adjusting their shape slightly when the substrate binds. This is called the induced fit model, where the enzyme molds itself around the substrate, creating an even more perfect fit. In that moment, both enzyme and substrate change shape slightly, as though bowing toward one another in mutual recognition. The adjustment lowers the activation energy further, ensuring that the reaction proceeds smoothly. Enzymes also operate with exquisite control. They respond to the environment within the cell, to temperature, to acidity, to the presence of inhibitors or activators. A small change in these conditions can alter an enzyme's performance dramatically. Most enzymes function best within narrow limits, their activity falling off when the conditions drift too far. High temperatures, for example, can cause the delicate folding of an enzyme's protein structure to unravel, destroying its function. Similarly, extremes of pH can disrupt the electrical charges that hold its shape together. Yet within their ideal range, enzymes perform with astonishing efficiency, catalyzing reactions millions of times faster than they would occur otherwise. It is through enzymes that the great cycles of life are maintained: metabolism, growth, repair, and decay. In the human body alone, thousands of enzymes work together in perfect coordination. In digestion, enzymes break food into its smallest parts. Amylases split starches into sugars, proteases cut proteins into amino acids, and lipases dismantle fats. In respiration, enzymes help convert these molecules into usable energy, feeding the mitochondria that power every cell. Even in thought, enzymes control the synthesis and breakdown of neurotransmitters, ensuring that signals travel smoothly between neurons. Every emotion, every movement, every act of healing depends upon their presence. Within each enzyme lies a story of precision built over billions of years of evolution. The earliest forms of life, floating in ancient seas, relied on primitive enzymes to capture energy from their surroundings. Over time, these molecules grew more complex, more efficient, adapting to new environments and new forms of metabolism. The enzymes we carry within us today are the result of that long refinement, structures shaped by nature's patience to fit their roles with almost divine accuracy. Some are ancient, nearly identical to those found in the simplest bacteria, while others are unique to complex organisms, specialized for the needs of higher life. Together, they form an unbroken lineage of molecular craftsmanship, stretching back to the dawn of biology. The subtle power of enzymes extends far beyond the individual cell. In ecosystems, they drive the cycles that sustain the planet. In the soil, enzymes released by microbes break down fallen leaves and decaying matter, returning nutrients to the earth. In the oceans, they help phytoplankton capture carbon and sunlight, feeding the base of the food chain. Air, enzymes play quiet roles, guiding the reactions that shape our atmosphere. Life itself is an endless cascade of enzyme-driven transformations, each one feeding the next in a seamless, self-sustaining balance. Yet perhaps the most astonishing thing about enzymes is their silence. They act without fanfare, without visible sign of motion. Within the cell, they move at incredible speed, yet all this activity unfolds in perfect calm. A single enzyme can catalyze thousands of reactions every second, yet it is neither consumed nor diminished. It simply continues, working tirelessly. If we could listen closely enough, perhaps we would hear the hum of their motion, the soundless music of transformation, the quiet energy of existence renewing itself. Even as enzymes perform their work, they are part of a delicate balance. Their activity must be regulated, neither too much nor too little. Cells control enzymes through feedback loops, subtle mechanisms that maintain equilibrium. When the product of a reaction becomes abundant, it may bind to the enzyme that created it, slowing its activity. When the product is scarce, the inhibition lifts and the reaction accelerates again. This self-regulation ensures that energy is not wasted and that the flow of metabolism remains steady. It is another example of life's gentle intelligence, the way order arises naturally from interaction and feedback. For all their complexity, enzymes remind us of a simple truth. Life thrives through cooperation. Each enzyme, like each person, plays its role within a greater whole. Alone, it can do little. Together, countless enzymes form a living network of reactions that sustain every form of being. They are the threads that connect all life, from the smallest microbe to the largest tree, from the pulse of a hummingbird to the steady beat of your own heart. As you rest and breathe, imagine this silent world within you. Thousands of enzymes moving, shaping, and transforming, breaking, building, and renewing with every heartbeat. They work without pause, without fatigue, performing the same graceful motions they have for billions of years. Their quiet diligence is what keeps you alive, what allows the warmth of your body, the clarity of your thoughts, the rhythm of your sleep. Life's complexity may seem vast, but at its core lies simplicity, the dance of molecules guided by unseen forces of balance and purpose. Enzymes are the dancers, their movements subtle and precise, their performance unending. They are the quiet catalysts of life, turning chemistry into vitality, transforming the stillness of atoms into the living harmony that breathes within you now. 8. Structural proteins, the framework of being. Every living thing, from the tallest tree to the smallest cell, owes its strength and form to a quiet but powerful class of molecules known as structural proteins. These are the builders, the weavers, and the architects of the biological world. They do not rush or react like enzymes. Instead, they endure. They give bones their strength, skin its elasticity, hair its texture, and muscles their resilience. They are the unseen framework upon which all living form depends. Without them, life would have no shape, no stability, and no ability to move gracefully through the world. Structural proteins are the scaffolding of existence, the still, patient counterpart to the restless chemistry that animates life. To imagine their work, think of a vast building, the bones of a cathedral, the beams of a bridge, or the strands of silk in a spider's web. These materials are built to last, designed to hold weight, to bend without breaking, and to return to their form when disturbed. The same principle governs the proteins that hold our bodies together. They form long, fibrous structures that twist, coil, and interlock to create tissues both flexible and strong. Where enzymes are compact and globular, built for precision, structural proteins are elongated and robust, built for endurance. Their role is not to catalyze reactions, but to provide the physical foundation on which all other biological activity takes place. Among these, collagen stands as the most abundant and remarkable. It forms nearly a third of all the protein in the human body and is found in skin, tendons, cartilage, and bone. Under a microscope, collagen appears as slender triple helices. Each chain is rich in amino acids such as glycine and proline, whose small, compact shapes allow the structure to twist tightly without strain. The result is a molecule of extraordinary tensile strength, one that can withstand stretching without snapping. In your body, collagen fibers assemble into bundles, creating tissues that can flex and resist pressure. When you smile, when you walk, when you breathe, the quiet resilience of collagen allows your tissues to stretch and return, maintaining both form and function. Over time, collagen also tells the story of age. In youth, it is plentiful and supple, weaving through the skin and joints like silk. As years pass, its production slows, and the fibers grow stiffer, leading to wrinkles and reduced elasticity. Yet even in its gradual decline, collagen remains the body's quiet companion, preserving structure as long as it can. It is a testament to nature's artistry that something so simple in design, a repeating triple helix, could provide the foundation for so much strength and grace. Another structural marvel is keratin, a tough, fibrous protein that forms hair, nails, feathers, claws, and the outer layer of skin. Like collagen, it is built for durability, but its composition gives it a different quality resistance to water and wear. The strength of keratin comes from its internal bonds, particularly disulfide bridges formed between sulfur-containing amino acids such as cysteine. These bonds act as crosslinks, connecting neighboring strands and preventing them from unraveling. The more disulfide bonds present, the harder the material. Hair and feathers, which must bend and move, have fewer of these crosslinks, while nails and hooves, which must resist pressure, contain more. It is in these details that nature achieves its quiet balance, fine-tuning the chemistry of proteins to match the needs of the living world. The formation of keratin-based structures also reveals a beautiful harmony between molecular order and biological purpose. Within each strand of hair or feather lies a matrix of keratin filaments arranged with near-crystalline precision. Under light, these arrangements scatter reflections, producing the subtle sheen of silk, the shimmer of feathers, or the luster of healthy hair. This interplay of structure and light shows that even the materials of strength can possess elegance, that guard the body's surface against wear and weather also contribute to its beauty, merging function with form in the quiet perfection of evolution. Another family of structural proteins, known as elastins, provides the gift of flexibility. Where collagen lends strength and firmness, elastin offers stretch and recoil. It is found in tissues that must move constantly, the lungs that expand and contract, the arteries that pulse with each heartbeat, the skin that bends and straightens with every gesture. Elastin molecules are arranged in loose, coiling chains that can extend when pulled and spring back when released. Their unique composition allows them to endure countless cycles of stretching without fatigue. The presence of elastin gives living tissue a kind of memory, the ability to return to its original shape after deformation, a property that is both mechanical and deeply poetic. Muscle, too, owes its power to structural proteins. Within each muscle fiber lie filaments of actin and myosin, sliding past each other in coordinated motion. Together, they form the machinery of contraction, translating chemical energy into movement. Actin provides the track, while myosin acts as the motor, pulling on the actin filaments to shorten the muscle. Though these proteins are functional and dynamic, they are also structural, creating the lattice that gives muscle its shape and strength. When viewed under a microscope, the repeating pattern of actin and myosin filaments creates the striped appearance of skeletal muscle, a reminder that even motion has structure and that every movement we make arises from ordered design. Beyond the human body, structural proteins shape the world of other organisms in astonishing ways. Insects spin silk threads composed of fibroin, a protein both strong and flexible, capable of forming webs that can withstand wind and rain. The silk of a spider, molecule for molecule, is stronger than steel and yet so light that a single strand can drift effortlessly in the air. Birds rely on keratin to form feathers, lightweight, aerodynamic structures that make flight possible. In corals, sponges, and shells, structural proteins combine with minerals to create frameworks of incredible intricacy. Across every species, these molecules appear again and again, adjusted in composition but united in purpose: to build, to protect, to endure. The creation of structural proteins begins in the same way as all proteins, as chains of amino acids assembled by ribosomes within the cell. But their journey continues long after synthesis. Many undergo modifications that prepare them for strength. Collagen, for example, must be processed through a series of enzymatic steps before its triple helices can form. Other structural proteins are exported outside the cell, where they assemble into fibers or matrices that support entire tissues. This process is slow and deliberate, guided by enzymes, ions, and the surrounding environment. It mirrors the patient construction of natural materials, the slow crystallization of minerals, the growth of trees, the layering of coral reefs. Life builds itself in calm persistence, one bond at a time. Even as they appear solid and unchanging, structural proteins are not static. They are constantly renewed, broken down, and rebuilt in cycles of repair. The collagen in your bones today is not the same collagen you carried a decade ago. This quiet turnover ensures resilience, allowing life to maintain its structure even as its components age and decay. In every living thing, destruction and creation coexist in balance, the old making way for the new, yet the form remaining the same. To think of structural proteins is to think of stability itself, of the quiet strength that holds the living world together. They are not flamboyant or reactive. They do not flash or flare. They simply endure, woven into every layer of existence. In their silence lies power, the strength of the spider's thread, the softness of skin, the resilience of a beating heart. They remind us that strength is not the opposite of gentleness, but its companion. It is the stillness beneath movement, the calm beneath life's constant change. As you rest and picture the world within you, imagine the fine network of structural proteins stretching from cell to cell, forming tissues, organs, and the graceful architecture of your body. See the collagen fibers like soft ropes of light, the keratin strands glimmering with quiet durability, the elastin coils bending and rebounding with each breath. They are the framework of being, supporting you in every motion and holding you together even in stillness. In their presence, life takes form, and in their endurance, it finds stability. 9. Transport and communication proteins. Inside every living being, there exists a delicate network of exchange, a constant movement of messages and materials that keeps the organism alive, balanced, and aware. The body, at every level, is a community of cells that must cooperate to function as a whole. Each cell has its own boundaries, yet none can exist in isolation. Oxygen must reach distant tissues, nutrients must find their way to cells in need, and information must flow like a soft current through the body's interior. The quiet miracle that makes this possible is the work of transport and communication proteins, the carriers, messengers, and interpreters of the living world. They form the invisible bridges that link every cell to its neighbors, every organ to the rest of the body, and every living thing to the greater harmony of life. The story of transport proteins begins in the bloodstream, the vast liquid river that flows through the body's hidden channels. Within this gentle current move the molecules of life: oxygen, carbon dioxide, nutrients, hormones, and countless others. Most of these molecules cannot travel freely. They are too reactive, too large, or too insoluble in water. They need carriers, guides that can hold them safely and deliver them to their destinations. Among the most beautiful of these carriers is hemoglobin, a protein that gives blood its deep red color. Hemoglobin is a marvel of molecular design. Each of its 4 subunits contains a heme group, a ring-shaped structure that holds an atom of iron at its center. It is this iron that binds oxygen, allowing the molecule to carry it from the lungs to the tissues. When you inhale, oxygen enters the lungs and binds to the hemoglobin in your red blood cells. Then, in the return journey, it picks up carbon dioxide, carrying it back to the lungs for release. The entire process is rhythmic and effortless, continuing from your first breath to your last. What makes hemoglobin so extraordinary is its sensitivity. It does not cling to oxygen too tightly, nor does it release it too soon. Instead, it responds to its environment to changes in pH, temperature, and the presence of carbon dioxide. In active tissues, where oxygen is consumed rapidly and carbon dioxide levels rise, hemoglobin changes shape, loosening its grip and freeing its oxygen. In the cooler, less active tissues, it holds on more firmly. This adaptability ensures that every part of the body receives what it needs, when it needs it, maintaining balance with perfect subtlety. Other transport proteins serve equally vital roles, each designed for a particular task. In the bloodstream, albumin carries fatty acids, hormones. Transferrin carries iron safely through the plasma, protecting the body from the reactive power of free iron ions. Lipoproteins ferry cholesterol and triglycerides through the watery medium of blood, forming the quiet link between the fats of the diet and the energy of the cell. Inside each cell, transport proteins move materials across membranes, delicate barriers that separate one compartment from another. These membrane transporters come in many forms. Some act as channels, forming small tunnels through which ions can flow freely. Others act as pumps, using energy to move molecules against concentration gradients. The sodium-potassium pump, for instance, is one of the most essential. It maintains the electrical balance across cell membranes by exchanging sodium ions for potassium ions in a steady rhythm. This subtle movement of charged particles creates the voltage that allows nerve cells to fire and muscles to contract. Transport proteins also carry small molecules inside the cell itself, distributing nutrients, metabolites, and waste products. Within the mitochondria, transporters move molecules like pyruvate and ATP, coordinating the exchange of energy between compartments. Even within the smallest organelles, transport proteins ensure that every reaction occurs in the right place and that no resource is wasted. It is as though the cell contains a system of unseen highways and gates, each managed by its own protein guardian, directing traffic with precision and care. But transport alone is not enough. For life to flourish, cells must also communicate, sending signals, receiving information, and adjusting to changes both within and without. Here, communication proteins take their place. They form the language of the cell, a language written not in words, but in shapes, charges, and molecular movements. On the surface of every cell lie proteins that act as receptors, the eyes and ears of the cell, constantly sensing the environment. When a signal arrives, often in the form of a hormone or neurotransmitter, it binds to its receptor like a key turning in a lock. This binding triggers a change in the present receptor's shape, a small but profound transformation that sends a message inward. The signal might open an ion channel, allowing calcium or sodium to flow in, or it might activate an enzyme inside the cell, setting off a cascade of reactions that alter metabolism, gene expression, or cell behavior. Through this elegant system, cells respond instantly to the needs of the body, coordinating every function from heartbeat to memory. One of the most familiar communication proteins is the insulin receptor. When you eat, glucose enters the bloodstream, and the pancreas releases insulin, a small protein hormone. Insulin binds to receptors on the surface of muscle and fat cells, instructing them to absorb glucose and use it for energy. The receptor, upon binding, activates a chain of intracellular signals that cause transporter proteins to move to the cell membrane, opening the way for glucose to enter. In this single act of communication, countless cells synchronize, balancing the body's energy levels and maintaining homeostasis. Communication proteins are also the foundation of the nervous system, where signals must travel with both speed and accuracy. In neurons, specialized proteins called ion channels open and close in response to changes in voltage or chemical signals, allowing ions to flow across the membrane. This flow creates electrical impulses, tiny waves of charge that travel along the nerve fiber. At the synapse, where one neuron meets another, neurotransmitters are released, crossing the narrow gap between cells. On the far side, receptor proteins receive these molecules, converting the chemical message back into an electrical one. This is how thought moves, how memory forms, how motion begins, a cascade of proteins passing messages in silence, faster than the blink of an eye. Even between distant cells, communication proteins maintain connection. Hormones, like adrenaline, growth factors, and cytokines circulate through the bloodstream, carrying information to far-off tissues. Each finds its specific receptor, delivering its message with accuracy. The cells that receive the signal respond appropriately. Some begin to divide, others slow their metabolism, others change the proteins they produce. It is as if the entire body listens to a single, soft symphony, each instrument playing its part at precisely the right moment. Transport and communication proteins work together as two halves of one system: movement and message, structure and signal. The transporters deliver the materials that make life possible. The communicators ensure that those materials are used in harmony. Their cooperation sustains every heartbeat, every breath, every thought. They maintain the invisible order that keeps life balanced, adapting fluidly to the ever-changing conditions of existence. If we could see this network as it truly is, we might glimpse a shimmering web stretching through every cell and vessel, every tissue and organ. Molecules pass along it like notes in a melody: oxygen, glucose, ions, hormones, each carried by a specific protein, each received by another. The flow never stops. It continues even as you rest, as you dream. It is the deep pulse of life itself, the molecular rhythm that has sustained every organism since the beginning. As you breathe now, hemoglobin carries oxygen through your blood. As your heart beats, ion channels open and close, guiding the electric waves that sustain your pulse. As your mind drifts toward rest, neurotransmitters pass their messages gently between neurons. All of it happens without effort or awareness, guided by proteins that neither think nor sleep, yet perform their roles with unwavering precision. In this quiet awareness, one can feel the unity of life, the way every motion, every signal, every breath is part of one seamless conversation, transport, and communication. Proteins are the translators of this conversation, turning chemistry into coherence, movement into meaning. Through them, the body speaks to itself in silence, a language older than thought and deeper than sound, a language that will continue to flow, softly and endlessly, as long as life endures. 10. Hormones and signaling molecules. In the vast and intricate world within the human body, every cell must know when to act, when to rest, and when to change its function. There are trillions of cells, each with its own role, yet all must work together in harmony. The signals that guide this symphony are carried by molecules so small and light that they drift through the blood like whispers, hormones, and other signaling molecules. These messengers form the body's internal language of communication, linking distant organs and coordinating countless processes. Through them, the mind speaks to the body speaks to itself, and life sustains its gentle rhythm from moment to moment. Hormones are not like the structural or enzymatic proteins that build and catalyze. They are communicators, transient and precise, carrying information rather than mass. Released by specialized glands, they travel through the bloodstream, finding their way to specific target cells, where they bind to receptor proteins that recognize them alone. Once received, the message they carry transforms the cell's behavior, sometimes within seconds, sometimes over hours or days. Some instruct cells to divide, others to slow down. Some control hunger, others influence mood, growth, or reproduction. Each one, though microscopic, shapes the entire organism. The endocrine system, which produces and manages these hormones, is one of the body's most delicate networks. Its glands, the thyroid, adrenal, pancreas, pituitary, and others are small but powerful, ripple through the body. A single drop of hormone-rich blood contains instructions for thousands of cells. The pituitary gland, often called the master gland, sits at the base of the brain and orchestrates many of these signals. It releases hormones that tell other glands when to act, stimulating the thyroid to release hormones that regulate metabolism, or the adrenal glands to secrete cortisol in response to stress. It is a hierarchy built on trust and balance, where each component listens to the others, ensuring the body's inner environment remains stable no matter the world outside. Among the most well-known hormones is insulin, a small protein that regulates blood sugar levels. After eating, glucose from digested food enters the bloodstream, raising its concentration. The pancreas senses this rise and releases insulin, which travels through the blood to cells in muscle, liver, and fat tissue. There, insulin binds to its receptor, a complex protein embedded in the cell membrane, and unlocks channels that allow glucose to enter. The cell absorbs the sugar and uses it for energy or stores it for later use. As glucose levels fall, the pancreas reduces its secretion, maintaining equilibrium. Without this system, the body's energy supply would fluctuate wildly, leading to exhaustion and damage. In people with diabetes, where insulin production or function is impaired, another vital hormone, adrenaline, illustrates the body's ability to react instantly to changing circumstances. Produced by the adrenal glands in moments of stress or excitement, adrenaline rushes through the bloodstream, preparing the body for action. It increases heart rate, dilates airways, and redirects blood flow toward the muscles. At the same time, it triggers the breakdown of glycogen in the liver, releasing glucose for quick energy. Within seconds, the body is alert and ready, a silent transformation driven by a molecule that vanishes as quickly as it appears. When the moment passes, other hormones, such as cortisol, take over, guiding the body back toward calm and recovery. Hormones also shape the slow, steady changes of growth and development. Human growth hormone, released by the pituitary gland, stimulates tissues to increase in size and repair themselves. During childhood and adolescence, it orchestrates the expansion of bones, muscles, and organs, ensuring the body grows in proportion. Thyroid hormones, produced in the neck, control metabolism, the rate at which every cell converts nutrients into energy. Even a slight imbalance can shift the entire body's rhythm. Too much thyroid hormone leads to restlessness and heat, too little to fatigue and chill. Each hormone's level is regulated through feedback loops, gentle checks and balances that prevent excess or deficiency. The brain monitors hormone concentrations in the blood and adjusts gland activity accordingly, maintaining a balance as precise as that of a spinning gyroscope. Some hormones influence not the body's structure, but the very essence of behavior and emotion. Deep within the brain, glands release molecules like oxytocin, serotonin, and dopamine chemicals that shape how we feel, connect, and experience the world. Oxytocin, often called the bonding hormone, is released during moments of closeness, trust, or affection. It strengthens relationships between parents and children, partners. Serotonin, on the other hand, regulates mood and calmness, influencing sleep, appetite, and contentment. Dopamine rewards us for actions that sustain life: eating, learning, achieving, connecting, reinforcing patterns of joy and motivation. These are not abstract ideas, but chemical realities, signals carried by molecules moving through the body's intricate pathways, connecting mind to matter. Even at the cellular level, signaling molecules coordinate life's smallest details. Not all of them travel through the blood. Some move only between neighboring cells, creating local networks of communication. Neurotransmitters, such as acetylcholine and glutamate, pass between neurons in the brain and nervous system. When an electrical impulse reaches the end of a neuron, these molecules are released into the tiny gap between cells. The entire brain, with its vast landscape of thoughts and sensations, operates through this microscopic exchange, a dance of molecules across spaces so small they could fit millions of times within a single grain of sand. There are also signaling molecules that do not move at all, but remain anchored in cell membranes, communicating through direct contact. When one cell touches another, these bound molecules recognize and respond, guiding development and immune defense. The immune system, for example, uses such signaling to distinguish self from non-self, allowing the body to recognize invading pathogens without attacking its own tissues. Cytokines, another family of signaling molecules, travel short distances to coordinate the immune response, calling cells to action when infection occurs and calming them once the danger has passed. The precision of these signals ensures that defense does not become destruction, that protection remains gentle even in its strength. In plants, too, signaling molecules perform miracles of coordination. Hormones like auxin and ethylene guide growth, flowering, and ripening. When a seed germinates, these molecules direct the young stem toward light and roots toward water. When fruit ripens, ethylene spreads through the air, signaling neighboring fruits to follow suit. Even in silence, the language of molecules connects one living thing to another, carrying meaning across invisible boundaries. In every organism, from the simplest to the most complex, hormones and signaling molecules weave life into coherence. They allow distant parts to act as one, responding to both internal needs and external changes. They ensure that growth, energy, defense, and reproduction occur not as random events, but as parts of a larger pattern. Their presence makes the body not merely a collection of cells, but a unified whole, a living conversation between molecules. If you were to visualize this molecular communication, you might see a landscape of glowing particles drifting through the bloodstream, each carrying its unique message. Some move quickly, reaching their targets in seconds. Others travel slowly, affecting the body over days or weeks. Around them, receptors wait like open hands, ready to receive what belongs to them alone. The molecules touch, connect, and release, sending waves of meaning through tissues and organs. It is a communication so subtle that it happens without sound or thought, yet so powerful that it shapes every moment of life. Even now, as you breathe and drift in calm awareness, countless messages are flowing through you. Hormones adjust your heart rate, regulate your temperature, guide your metabolism, and soothe your mind. Signaling molecules move across the tiny spaces in your brain, carrying thoughts and memories from one cell to the next. The entire body listens and responds, in perfect synchrony, maintaining balance and peace. In this harmony of signals lies the essence of life: connection without noise, communication without words. Hormones and signaling molecules are the silent language of being, carrying the intelligence of nature through every corner of the body. Through them, the body knows itself, the mind touches matter, and existence becomes awareness. As you rest, this quiet symphony continues within you, steady and sure, a molecular whisper that keeps the rhythm of life alive. 11. The synthesis of proteins inside the cell. Within every living cell, hidden from sight, lies a world of motion and precision, where the fundamental process of life unfolds: the synthesis of proteins. It is a process that happens quietly, without pause, and with astonishing coordination. From the simplest bacterium to the most complex human body, this act of creation sustains existence itself. Every structure, every enzyme, every signal that moves through the body begins with the making of proteins. It is here, in the soft hum of cellular machinery, that the code of life, written in DNA, is transformed into tangible form, a chain of amino acids that will fold, interact, and come alive as the molecules that make all living things possible. To imagine this process, picture a cell not as a static entity, but as a vast ocean filled with floating structures, each performing its task in perfect time. Within this ocean lies the nucleus, a protected chamber that holds the genetic instructions, the DNA, the great library of life. Every protein that will ever exist begins as a sequence of genetic information, a specific order of nucleotides arranged like letters in a long and ancient text. This genetic script does not move freely from the nucleus, for it must remain safe and unaltered. Instead, the cell makes a copy, a single-stranded transcript of the required section of DNA, known as messenger RNA, or mRNA. This is the first step of protein synthesis. Transcription. The careful rewriting of information from one language to another. During transcription, an enzyme called RNA polymerase glides along the DNA strand, reading its sequence, one base at a time. For every base it encounters adenine, thymine, cytosine, or guanine, it assembles a complementary RNA nucleotide. Where DNA has thymine, RNA uses uracil, a subtle but significant difference. Slowly, the mRNA strand grows, its sequence mirroring that of the DNA template. When the enzyme reaches the end of the gene, the mRNA detaches, carrying the precise blueprint for a single protein. It is as if a page has been copied from the library of life and handed to a messenger, ready to be delivered to the cell's workshops. The mRNA leaves the nucleus through small openings in the nuclear envelope, entering the cytoplasm where ribosomes await. Ribosomes are the factories of protein synthesis, tiny, complex structures made of ribosomal RNA and proteins. Some float freely in the cytoplasm, producing proteins that function inside the cell. Others are attached to the rough endoplasmic reticulum, where they synthesize proteins destined for export or use in membranes. Wherever they are, their purpose is the same: to read the mRNA and translate its code into a chain of amino acids, each precisely chosen and placed. This next phase is called translation, the conversion of genetic information into the language of life's chemistry. The ribosome binds to the mRNA, scanning its sequence in sets of 3 bases called codons. Each codon corresponds to a specific amino acid, and this relationship, known as the genetic code, is universal across all known life. It is the language that unites all organisms, from single-celled bacteria to the human being reading these words. To interpret this code, the ribosome relies on another kind of molecule, transfer RNA, or tRNA. Each tRNA carries a specific amino acid and has a region called an anticodon, which pairs perfectly with its corresponding codon on the mRNA. As the ribosome moves along the mRNA, tRNAs arrive one by one. Guided by this pairing, the amino acid from each tRNA is transferred to the growing chain, linked to the next by a peptide bond. This process is rhythmic, patient, and continuous. The ribosome acts as both a translator and a builder, ensuring that the amino acids are assembled in the exact sequence dictated by the mRNA. With every new bond formed, the chain grows longer, extending outward like a thread being woven into a fabric of life. The accuracy of this process is almost miraculous. The ribosome can read thousands of codons without error, ensuring that each amino acid finds its proper place. If even a single amino acid were misplaced, the resulting protein might fold incorrectly or lose its function entirely. To prevent such errors, the cell employs elaborate proofreading mechanisms. Certain enzymes check that each tRNA carries the correct amino acid before it participates in synthesis, and the ribosome itself verifies the pairing between codon and anticodon. This meticulous care reflects the precision upon which life depends, a quiet vigilance that ensures harmony in the midst of ceaseless motion. As the chain of amino acids lengthens, it begins to fold upon itself, even before the synthesis is complete. The sequence of amino acids determines how it will bend and twist, guided by the forces between atoms and side chains. In some cases, helper proteins known as chaperones assist in this process, preventing premature folding or aggregation. When the ribosome reaches a special codon that signals the end of the sequence, it releases the completed chain, a new protein, born into the cell's interior. What began as a line of code within DNA has become a living molecule, ready to perform its role in the symphony of life. Many proteins require further refinement before they become fully functional. Some fold into complex three-dimensional shapes, stabilized by internal bonds. Others are modified by the addition of small chemical groups or sugars, processes that occur within the endoplasmic reticulum and the Golgi apparatus. These modifications determine where the protein will go and what it will do, whether it will remain within the cell, form part of the membrane, or be secreted into the bloodstream to act elsewhere. The cell is meticulous in these preparations, sorting and packaging each protein with care, sending it on its way like a message addressed to a specific destination. It is humbling to consider how many of these tiny factories are at work at any given moment. In a single human cell, thousands of ribosomes operate simultaneously, producing millions of proteins every hour. Each one follows its instructions with unwavering precision, maintaining the continuity of life without conscious guidance. It is an orchestration of unimaginable scale and subtlety, all occurring within the confines of a space so small that it cannot be seen by the naked eye. This process, transcription, translation, folding, and modification, has remained unchanged for billions of years. It is one of the most ancient patterns in existence, the same in every creature that has ever lived. From the first self-replicating cells in the primordial oceans to the complex multicellular organisms of today, protein synthesis has carried life forward, linking generation to generation. The DNA that directs it has changed through time, but the language remains the same. Its grammar universal, its message enduring. As you imagine this process, picture the interior of a cell as a place of quiet creation. The DNA rests like a sacred text in its chamber. RNA polymerase moves along it, tracing lines of information. The messenger RNA drifts outward, a ribbon of instruction carried by invisible currents. The ribosome receives it, the tRNAs arrive in order, and amino acids join one another in a soft, steady rhythm. The chain folds, a shape emerges, and a new protein is born. It may become an enzyme that drives a reaction, a structural element that supports the cell, or a messenger that carries information. Whatever its role, it joins the great cycle of life, a small part of a vast, interconnected whole. Even as you rest and breathe, this process continues within you, ceaseless and unseen. Every heartbeat, every breath, every renewal of tissue depends upon it. Proteins are synthesized, used, and replaced in endless succession. The body is never static. It is a work in progress, continually rebuilt by the very molecules it creates. The synthesis of proteins is not just a process of chemistry. It is an act of persistence, of continuity, of life affirming itself again and again. In the stillness of this understanding, one can feel a quiet reverence for the harmony that sustains existence. Every cell in your body knows what to do. Every strand of DNA holds the memory of ages, and within each ribosome, the ancient song of life is still being sung, wordless, tireless, eternal as the language of creation continues to echo through every living thing. 12. Denaturation. When proteins lose their shape, in the serene harmony of life's inner workings, every molecule has its place and purpose. Proteins, the quiet laborers of biology, exist in countless forms: enzymes, structural fibers, receptors, carriers, each perfectly folded to perform its role. Their shapes are not arbitrary. They are the result of delicate forces, balanced precisely to create the curves, folds, and bonds that give them function. Yet, like all things in nature, proteins are not indestructible. They live in a fragile balance, and under certain conditions, that balance can be broken. When the forces that hold a protein's shape are disturbed, it unfolds, losing its structure and, with it, its function. This process is called denaturation. Though invisible to the eye, It is one of life's quiet reminders that form and function are one and the same, that even the smallest disruption can ripple through the grand architecture of being. A protein's shape is its soul. Each one begins as a simple chain of amino acids, linked together by peptide bonds in a specific sequence. But the chain does not remain straight. It folds into loops, spirals, and sheets, guided by the attractions and repulsions between its atoms. These interactions, hydrogen bonds, ionic connections, hydrophobic forces, and van der Waals attractions, create the secondary, tertiary, and quaternary structures that define the protein's three-dimensional form. Within that form lies its ability to function, to bind to other molecules, to catalyze reactions, to transmit signals, or to support structures. When that form is disrupted, when the folds loosen and the bonds break, the protein cannot do what it was made to do. Denaturation is, therefore, both a physical and a symbolic transformation, a return from order to simplicity, from function to silence. In most cases, denaturation occurs when the environment around the protein changes beyond its tolerance. Temperature, pH, and chemical conditions all influence the fragile network of forces that maintain structure. Heat, for instance, is one of the most common agents of denaturation. As temperature rises, atoms within the protein vibrate more intensely, and the delicate hydrogen bonds that hold helices and sheets together begin to break. At first, the protein may quiver, but remain intact. But as the energy increases, the bonds unravel one by one until the entire structure collapses. What was once a precisely folded molecule becomes an uncoiled thread, unable to return to its former shape. This is why the clear liquid of an egg turns white and opaque when cooked to proteins in the egg white, primarily albumin, denature, and aggregate, forming a new, solid structure. The process is irreversible, and in its simplicity, it demonstrates the profound power of molecular form. Changes in acidity can cause a similar effect. Proteins depend on the balance of electrical charges within their structure, positive and negative regions that attract and stabilize each other. When the pH shifts, the distribution of these charges changes. Bonds that once held the structure firm may repel each other instead. A drop of lemon juice, curdling milk, or stomach acid unraveling dietary proteins during digestion are gentle examples of this process in action. Denaturation is not always destructive. In digestion, it is essential. The proteins in food must be unfolded before enzymes can break them down into amino acids, allowing the body to absorb and reuse them in the synthesis of new proteins. In this way, denaturation is both an ending and a beginning, the dissolution of one form to make room for another. Chemical agents can also denature proteins, particularly those that interfere with hydrogen bonding or hydrophobic interactions. Alcohol, detergents, and heavy metals are examples of such agents. Alcohol, used as a disinfectant, denatures the proteins in bacterial membranes, disrupting their function and causing the cells to die. Detergents, though gentler, work in a similar way. Heavy metals like mercury or lead bind tightly to the sulfur atoms of amino acids, altering their shape and preventing normal folding. These processes reveal the vulnerability of proteins, how easily the balance of life can be disturbed by chemical forces that lie outside the natural range. Despite its fragility, denaturation can sometimes be reversed. In mild cases, when the external stress is removed, some proteins refold into their native structure, regaining their function. This ability, known as renaturation, depends on whether the primary structure, the sequence of amino acids, remains intact. If the peptide bonds themselves are unharmed, the protein may remember its shape and refold spontaneously, guided by the same forces that shaped it originally. This self-restoring capacity illustrates a kind of molecular memory, a resilience that allows life to recover from disruption. But if the damage is too great, if the bonds are broken or the environment remains hostile, the protein's shape is lost forever. Denaturation does not always signify destruction in the grand scheme of life. Often, it is part of the natural rhythm of change and renewal. Within the body, old or damaged, proteins are regularly unfolded and degraded by specialized systems. Enzymes, called proteases, recognize these denatured proteins and break them down into their component amino acids, which are then reused to make new ones. In this way, denaturation becomes part of life's quiet recycling, a continuous flow of transformation that ensures balance and efficiency. Even in death, the molecular essence persists, ready to be reshaped into new forms. Yet, when denaturation occurs where it should not within living cells, under stress or disease, the effects can be harmful. Misfolded or denatured proteins sometimes clump together, forming aggregates that the cell cannot easily remove. These aggregates can interfere with normal functions, leading to conditions such as Alzheimer's, Parkinson's, or prion diseases. In these cases, the protein's lost shape creates a chain reaction, causing neighboring molecules to misfold as well. It is a reminder that life's order depends on the delicate interplay of structure and environment. When that balance fails, the system falters, and chaos gently takes its place. Still, even this fragility has beauty in it. The fact that proteins can lose their form reminds us how dynamic life truly is, never static, never frozen in perfection. Every molecule exists in motion, constantly responding to changes in heat, chemistry, and energy. Denaturation, then, is not merely loss, but transformation. It mirrors the natural cycles of the world. The breaking down of old leaves to nourish new growth, the soft decay that returns matter to the soil. In the microscopic world, as in the larger one, if one were to observe denaturation at a molecular level, it might look like a slow unfurling, a release of tension. The folded protein, once compact and purposeful, begins to loosen, its coils relaxing one by one. Bonds slip away, and the structure opens into a fluid, flexible thread. In some environments, the thread tangles with others, forming new patterns, not living ones, but stable in their own quiet way. In others, it drifts apart completely, its parts returning to the molecular sea from which they came. It is an ending without violence, a dissolution into simplicity. Even now, as your body rests, denaturation and renaturation occur in balance. Proteins are made, used, unfolded, and replaced. Heat rises and falls, acidity shifts with digestion and breath, and cells manage these changes with unerring care. The harmony of life is not in unchanging order, but in the balance between structure and fluidity, between formation and decay. The folded protein and the unfolded one are both part of this cycle, both necessary to the quiet persistence of living matter. In the end, denaturation is a lesson in impermanence. It shows that form, no matter how intricate, is always subject to the forces that surround it. It reminds us that even the smallest shapes, those that sustain the breath and movement of life, are bound by the same laws as everything else, the laws of energy. Yet within that impermanence lies continuity, for what unravels will one day fold again, and what dissolves will find new purpose in another form. As you breathe and drift into calm, imagine the proteins within you, some perfectly folded, others slowly releasing their shapes. They are not failing. They are part of the eternal process by which life renews itself. The quiet unraveling, the soft return to simplicity, is not an ending but a transformation, one that mirrors the gentle truth of nature itself. Nothing is ever truly lost. It merely changes its shape, waiting patiently for the moment when creation begins again. 13. Nutritional perspective. Proteins in the human diet. Every living body, from the simplest organism to the most complex human form, depends upon a steady flow of nourishment to sustain its endless renewal. The cells that make up your muscles, organs, and tissues are not fixed or permanent. They are in a constant state of change, breaking down old materials, rebuilding new ones, adapting to the quiet rhythms of growth and repair. At the heart of this ongoing renewal lies one essential class of nutrients: proteins. They are the body's raw materials, the tools, and the structure of life itself. Each mouthful of food you take, each grain, fruit, or fragment of nourishment contains within it the potential to be transformed into part of you: your skin, your hair, your heartbeat, your thoughts. In this calm reflection on biology and sustenance, we journey through the gentle science of proteins in the human diet, exploring how the molecules of life sustain the body that hosts them. When you eat, you are not simply filling yourself with energy. You are participating in the exchange of matter that connects all living things. The proteins in food are composed of long chains of amino acids, the same fundamental building blocks that make up every protein within your own body. Yet before these amino acids can become part of you, they must be liberated from their existing forms. In the stomach, the process begins quietly. Gastric juices, rich in hydrochloric acid, unfold the complex proteins in food, denaturing them much like heat denatures the proteins in an egg as it cooks. This unfolding makes them accessible to enzymes such as pepsin, which begin cutting the long chains into smaller fragments called peptides. As the food moves into the small intestine, other enzymes, trypsin, chymotrypsin, and peptidases continue this work, slicing the peptides into individual amino acids. By the time digestion is complete, the food's proteins are no longer recognizable. They have been reduced to their simplest components, ready to be absorbed and reused in the construction of new proteins that belong uniquely to you. These amino acids enter the bloodstream through the lining of the intestine, and are carried to every cell in the body. There, they serve many purposes. Some are used immediately to build new proteins, enzymes that catalyze reactions, hormones that carry signals, or structural elements that form the body's tissues. Others are stored temporarily in what biologists call the amino acid pool, a reservoir that supplies the constant demand for rebuilding and maintenance. The body does not store protein in the way it stores fat or carbohydrates. Instead, it continuously recycles amino acids, breaking down old proteins to make new ones. This ceaseless exchange keeps you alive and whole, even as the matter that composes you is constantly changing. Of the 20 amino acids required by the body, 9 are called essential, meaning they cannot be synthesized from other compounds and must be obtained through food. These are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Without them, the machinery of life would slow and falter. The remaining amino acids are non-essential, not because they are unimportant, but because the body can produce them from other molecules. Together, they form a complete alphabet of chemistry that allows cells to write the countless proteins necessary for survival. The sources of dietary protein are varied, and each brings its own balance of amino acids. Animal-based proteins, such as those found in meat, fish, eggs, and dairy, are considered complete, containing all 9 essential amino acids in proportions similar to those the body requires. Plant-based proteins, by contrast, often lack one or more essential amino acids, yet they carry their own richness, providing fiber, minerals, and compounds that support health in other ways. When consumed in variety, plant proteins complement one another beautifully. Grains, for example, are lower in lysine but rich in methionine. Together, they form a complete protein source, a partnership as old as agriculture itself. Across cultures, traditional cuisines naturally reflect this balance. Rice and beans, corn and lentils, bread and hummus, simple combinations that quietly fulfill the body's intricate molecular needs. Beyond composition, The quality of protein also depends on its digestibility, how efficiently the body can break it down and absorb its amino acids. Cooking generally enhances digestibility, as heat denatures the protein, making it more accessible to enzymes. Fermentation, too, can improve bioavailability by partially breaking proteins down before consumption. In every culture, such techniques have evolved intuitively, long before their biochemical foundations were understood. They reveal the deep connection between the art of nourishment and the science of life, both guided by the same natural wisdom. Protein requirements vary with age, activity, and circumstance. Infants and children need more relative to their body weight, for their cells are growing rapidly, dividing and forming tissues at a tremendous rate. Adults require less for growth but still depend on steady intake to maintain muscles, repair tissues, and synthesize enzymes and hormones. During times of illness, stress, or healing, the demand for protein rises again as the body turns inward to rebuild itself. The recommended daily intake for an average adult is modest, roughly 0.8 grams per kilogram of body weight, yet even small shortages, sustained over time, can lead to fatigue, weakness, or impaired immunity. Conversely, excess protein, when far beyond what the body needs, is converted into energy or stored as fat, a reminder that balance, not abundance, sustains health. Beyond their structural role, proteins in the diet influence nearly every aspect of physiology. Certain amino acids act as precursors for neurotransmitters, the molecules that carry signals within the brain. Tryptophan, for example, is converted into serotonin, which promotes calmness and well-being. Tyrosine gives rise to dopamine and norepinephrine, which support focus and energy. Methionine contributes to the production of antioxidants that protect cells from damage. Through these pathways, the proteins in food subtly shape mood, cognition, and resilience. They nourish not only the body but the mind. It is humbling to realize that every meal is, in essence, a transfer of life's materials. The protein in a bean or a grain was once part of a living cell, performing its own functions before being offered to you through the chain of nourishment. When you eat, you take in the remnants of other lives, reassembling them into your own. This exchange is the heartbeat of ecology, energy, and matter moving in cycles, one life giving rise to another. The amino acids that build your muscles today may once have been part of the leaves of a plant, the body of a fish, or the grain of the earth. In this way, every living thing is connected through the shared currency of protein, each participating in the same unending conversation of creation and renewal. In moments of quiet reflection, one can see that the act of eating is both simple and sacred. Each bite begins a molecular journey that ends in the sustenance of life. Digestion, absorption, synthesis, all happen silently, faithfully, guided by the same forces that have operated since life began. The proteins you consume today will be broken down and rebuilt into the tissues that allow you to breathe. And when those tissues, in time, are replaced, their components will return to the cycle once more, becoming part of another life. If you imagine this process on the smallest scale, they may become an enzyme that catalyzes a reaction, a strand of collagen that strengthens the skin, or a molecule of hemoglobin that carries oxygen through your blood. Each one finds its place, guided by the pattern written in your DNA. The food you eat becomes you, transformed with perfect precision into the architecture of your being. In this awareness, there is peace. The body is not separate from the world. It is made of the same materials, following the same rules, sustained by the same cycles. Proteins in the diet are more than nutrients. They are bridges between self and nature, between one life and another. Every molecule carries a memory of sunlight, air, and earth. Every bite is a quiet act of connection, a renewal of the promise that life gives to itself. The act of nourishment so ordinary and yet so profound, is a reflection of the harmony that pervades all things. Within the smallest cell, the largest organism, and the universe itself, there is the same gentle rhythm, the taking in, the transformation, the giving back. Through the proteins you eat and the ones your body creates, you are part of that rhythm, part of the unbroken song of life. 14. The role of proteins in evolution and diversity. Across the immense history of life on Earth, from the first flicker of self-replicating molecules in ancient seas to the boundless variety of organisms that fill our planet today, one constant thread unites all forms of existence: the quiet and persistent work of proteins. These intricate molecules, assembled from simple amino acids, have been the artisans of evolution, shaping every living structure and guiding every adaptation that life has ever achieved. In their endless permutations, they carry both memory and possibility, the record of what has been and the potential for what may yet come. To understand the role of proteins in evolution is to see life itself as a long unfolding of molecular creativity, written in the language of chemistry and refined by the patient hand of time. In the earliest moments of life's history, when Earth's surface was still cooling and the oceans shimmered with the raw materials of creation, simple organic molecules drifted and collided in the warm, mineral-rich waters. Among them, chains of amino acids began to form, perhaps catalyzed by the heat of volcanic vents or the energy of sunlight. These primitive peptides were unstable and short-lived, yet they marked the beginning of something extraordinary. Over countless cycles of chance and selection, certain combinations proved more stable, more capable of interacting with their surroundings. Some may have bound to nucleic acids, assisting in replication or catalyzing chemical reactions. In those moments, the first partnership between proteins and genetic material was forged, a bond that would define all life thereafter. As time passed, evolution refined these early molecules into more complex forms. The relationship between DNA and proteins deepened, each depending on the other. DNA provided the instructions. Proteins executed them, building and maintaining the living cell. This interplay gave rise to the first true organisms, simple, single-celled beings, capable of sustaining themselves, dividing, and evolving. Within their membranes, proteins played countless roles, forming channels that allowed nutrients to enter, catalyzing reactions that produced energy, and providing scaffolds that gave shape and protection. With each new generation, random changes in the genetic code altered the sequence of amino acids, producing proteins with new properties. Most changes led nowhere, but some granted an advantage: a slightly faster enzyme, a sturdier membrane, a more efficient way to harvest light or store energy. Those subtle differences accumulated over eons, becoming the foundation of biological diversity. The flexibility of proteins is what allowed evolution to flourish. A small change in a gene could alter one amino acid in a protein, and that single substitution could transform its function. Sometimes, an enzyme that once acted on one molecule would begin to recognize another. A structural protein might gain elasticity or rigidity, allowing new forms to emerge. Through duplication and mutation, entire families of proteins evolved, diverging to perform specialized tasks. One ancestral enzyme might give rise to dozens of variants, each tuned to a different function. Over time, this branching of molecular lineages mirrored the branching of life itself, the diversification of species, each exploring a new niche in the grand experiment of nature. Consider hemoglobin, the protein that carries oxygen in our blood. Through countless small changes, these molecules evolved greater efficiency, allowing early animals to transport oxygen through their tissues and grow larger, more complex, and more active. The structure of hemoglobin, with its 4 interlocking subunits and iron-containing heme groups, is a masterpiece of molecular evolution, the product of billions of years of adaptation. Yet its variations across species reveal the ongoing dialogue between form and function. The hemoglobin of a deep-sea fish binds oxygen tightly in cold, low-oxygen waters, while that of a bird flying at high altitude releases it easily to sustain flight. Each version tells a story of environment, challenge, and survival. Proteins also enabled the rise of multicellular life, a turning point in the history of evolution. When single cells began to cooperate, they needed ways to adhere to one another, to communicate, and to divide labor. Structural proteins formed the glue that held them together, creating tissues. Communication proteins carried signals across membranes, coordinating behavior among neighboring cells. Enzymes managed the complex chemistry of growth and differentiation. This cooperation allowed new forms to emerge: filaments, sheets, organs. Eventually, entire organisms composed of billions of coordinated cells. Every muscle fiber, every nerve, every skin cell depends on proteins that evolved to connect, support, and guide them, transforming individuality into community. Even the diversity of life's outward appearance, the colors, textures, and movements that fill the natural world arises from the versatility of proteins. The iridescence of butterfly wings comes from structural proteins that bend light through microscopic patterns. The strength of spider silk, lighter than air yet stronger than steel, is due to proteins precisely aligned into crystalline strands. The glow of a jellyfish in the dark ocean is created by a single protein, green fluorescent protein, which captures and releases light in soft radiance. Through evolution, nature has painted itself with proteins, turning molecules into color, motion, and grace. Within the human body, the story continues on a smaller scale. The diversity of proteins among individuals contributes to our uniqueness. Tiny differences in DNA, a single change in a single gene, can alter a protein's shape or behavior, influencing everything from the color of one's eyes to the efficiency of metabolism. Some of these variations are neutral, others beneficial, and a few harmful, but together they form the tapestry of human diversity. They are the molecular echoes of our evolutionary journey, reminders that each of us carries within ourselves the record of 1 billion years of adaptation and change. At the level of populations, proteins serve as markers of ancestry and connection. Scientists can trace the lineage of species by comparing their protein sequences, finding patterns that reveal shared origins. The more similar two proteins are between organisms, the more recently their ancestors diverged. This molecular evidence confirms what fossils and anatomy have long suggested, that all life on Earth shares a common origin. From bacteria to plants to humans, we are all variations of the same molecular theme, shaped by the same biochemical vocabulary, speaking the same ancient language of amino acids. Yet evolution is not a finished process. Even now, proteins continue to change, adapting to new challenges and environments. In the presence of antibiotics, bacteria evolve enzymes that can neutralize the drugs. In the depths of the ocean, new forms of life develop proteins that function at crushing pressures and near freezing temperatures. In the desert, plants evolve heat-stable enzymes that continue to work in the glare of relentless sun. Each adaptation is written in the structure of a protein, an invisible reflection of the world that shaped it. When one considers this vast web of molecular evolution, it becomes clear that proteins are both the product and the driver of life's diversity. They are shaped by change, and in turn, create the capacity for change. Every new protein that emerges through mutation offers a possibility, a new reaction, a new structure, a new way for life to meet the world. Evolution, then, is not a process imposed from above, but one that arises from within, from the quiet restlessness of molecules seeking stability, function, and balance. If you were to imagine the evolutionary history of proteins, it would appear as a great branching tree. At each branch point, a new protein arises, a variation on an old theme, and from it, new forms unfold. The tree continues to grow, nourished by the flow of time and the constant testing of the environment. And within you, within every living cell, that same process still whispers, Every molecule carries not only the memory of its ancestors, but also the potential for what may come next. As you rest and contemplate this quiet continuity, consider that the proteins in your body are the living descendants of those first molecules that formed in the protein ancient oceans. Their sequences have changed, folded, and evolved, but the essence remains. You are built from the same chemistry that shaped the earliest forms of life, refined through unbroken generations of transformation. The diversity of life, the wings of a bird, the leaves of a tree. In the grand silence of nature, proteins are the storytellers, carrying within their forms the tale of life's beginning and its infinite unfolding. 15. Protein misfolding and molecular imperfection. In the quiet depths of every living cell, where molecules move with steady purpose, there exists an astonishing order, a choreography so precise that it sustains the rhythm of life without pause. Yet within this delicate balance, there are moments of imperfection, moments when the harmony falters and the flawless folding of proteins takes an unexpected turn. It is a reminder that even the smallest units of life are not immune to error, that within nature's precision there is room for fragility. Protein misfolding is one of these quiet deviations, a subtle but profound process in which a molecule loses its way, folding incorrectly and drifting from its intended path. Though often unseen, this imperfection holds deep significance. It reveals how closely creation and decay intertwine, and how even error can carry meaning within the fabric of existence. To understand misfolding, one must first recall how a protein comes into being. Each begins as a linear chain of amino acids, synthesized according to the instructions encoded in DNA. As the chain emerges from the ribosome, it folds upon itself, guided by chemical attractions, repulsions, and the fluid environment of the cell. Under ideal conditions, the forces of nature, hydrogen bonds, hydrophobic interactions, and electrostatic charges guide it gently into the correct shape. This process is swift and precise, a quiet miracle that occurs countless times each second within every organism. But the path to perfection is narrow, and even the slightest disturbance can lead a protein astray. A misplaced charge, a mutation in the genetic code, an excess of heat, or a deficiency of chaperone proteins can cause the molecule to fold incorrectly, creating a structure that no longer fits its purpose. A misfolded protein is like a poem with its words rearranged. The letters are all the same, but the meaning is lost. Its surfaces may expose parts that were meant to remain hidden. Its bonds may strain against one another, and its once stable form may tremble and collapse. Some misfolded proteins are harmless, quietly broken down by the cell's recycling systems and replaced with new ones. Others resist this fate, clinging to abnormal shapes that persist and accumulate. Over time, they may bind together, forming clusters or fibrils that disrupt the delicate environment of the cell. These aggregates are small, but their impact can be immense, leading to the slow deterioration of tissues and functions. In this way, a single molecule's mistake can echo through the living body, creating ripples that reach across time and health. Within the human body, protein misfolding lies at the heart of several profound diseases, conditions that remind us how fragile the harmony of life can be. In Alzheimer's disease, for instance, specific proteins such as amyloid beta and tau lose their proper structure and begin to aggregate within the brain. These clumps interfere with communication between neurons, gradually eroding memory, cognition, and identity. In Parkinson's disease, another protein called alpha-synuclein misfolds and accumulates, forming insoluble deposits that disrupt the smooth coordination of movement. In both cases, what begins as a small molecular imperfection becomes a cascade of dysfunction, spreading quietly from cell to cell. The brain, once an orchestra of perfect timing, begins to lose its rhythm, not through sudden violence, but through the slow unraveling of molecular order. There are other, even more haunting examples. Prion diseases such as Creutzfeldt-Jakob disease in humans and mad cow disease in cattle arise entirely from misfolded proteins that can transmit their shape to others. A single prion molecule, already folded incorrectly, can come into contact with a normal version of the same protein and cause it to misfold as well. This chain reaction continues, converting healthy proteins into the same distorted form, spreading like a whisper through the tissues of the brain. What makes prions so remarkable is that they carry no DNA, no RNA, no genetic material at all. They are pure form, pure pattern information stored not in a sequence but in a shape. It is as if the misfolded protein becomes a dark mirror of evolution, a self-replicating imperfection that turns life's order against itself. Yet even in these unsettling processes, there is a strange, almost poetic logic. Misfolding reminds us that life is not a static perfection, but a constant balancing act between stability and change. Proteins fold because they must, because function depends on form, but the same flexibility that allows them to fold also makes them vulnerable. Their ability to shift, to bend and twist into complex shapes, gives rise to the potential for error. Nature tolerates this risk because it is also the source of creativity. Without variation, without the chance of imperfection, evolution could not proceed. Every new function that life has ever discovered began as a variation in molecular structure, a subtle change, an altered fold, a mutation that led to something new. Thus, within the quiet tragedies of misfolding lies the same principle that makes life dynamic: the willingness of matter to experiment with itself. Inside the cell, there are guardians that protect against these molecular missteps. Chaperone proteins, named for their guiding role, help new proteins fold correctly and prevent them from tangling or clumping together. They patrol the crowded environment of the cytoplasm, rescuing those that stray and marking others for degradation if they cannot be saved. The proteasome, a complex molecular machine, acts as a recycling system, breaking down damaged or misfolded proteins into amino acids that can be reused. Lysosomes, too, play their part, enclosing and digesting aggregates before they grow too large. These systems keep the balance, ensuring that disorder does not overwhelm the cell's harmony. But like all defenses, they are finite. When the burden of misfolded proteins becomes too great through aging, genetic mutation, or environmental stress, the system falters, and the imperfections begin to accumulate. Aging itself may, in part, be the gradual consequence of this accumulation. Over time, as cells divide and renew themselves, the machinery of repair becomes less efficient. Misfolded proteins linger longer, forming small aggregates that the body can no longer clear away. These aggregates interfere with cell function, contributing to the slow decline in resilience that characterizes age. It is a process as natural as the fading of sunlight at dusk, not a failure, but a continuation of the same principles that govern all of nature. In the great cycle of creation and decay, even imperfection has its place, reminding us that time touches everything, even the molecules that make us. If one could observe protein misfolding at a molecular level, it would appear almost like a gentle unraveling. A protein, once folded into its perfect form, begins to quiver. Its hydrogen bonds break one by one. Its helices loosen, and its interior unfolds. Two misfolded strands drift close together, their sticky surfaces meeting and holding fast. Others join, forming a twisted filament that slowly grows. To the eye, it would be beautiful, a pattern of repeating structure, a crystalline order emerging from disorder. Yet this beauty carries with it the weight of dysfunction, for within the living cell, such aggregates block the pathways of life. They are molecular echoes of entropy, small reminders of the universal tendency toward disarray. And yet, in the broad perspective of existence, misfolding is not simply failure, it is part of the grand equilibrium. For every protein that misfolds, countless others fold correctly. For every error, the cell finds a way to adapt or renew. Life persists, not because it avoids imperfection, but because it integrates it into the cycle of being. The misfolded protein, once degraded, provides amino acids for new creations. The damaged cell gives way to another, stronger and more refined. This is how life endures through constant repair, constant reinvention, and the quiet acceptance of imperfection as part of the whole. As you rest and imagine this inner world, think of your cells as tiny universes, each maintaining its balance between order and disorder. Within them, proteins fold and refold, some achieving perfect form, others faltering and being gently taken apart. There is no judgment in this process, no failure, only transformation. Even the proteins that lose their way contribute to the greater rhythm of life, teaching the system how to adjust, how to survive, how to evolve. In the end, protein misfolding is both a symbol and a truth that perfection, though beautiful, the universe itself is built upon a balance between precision and change, and within that balance lies the possibility of life. In every moment of imperfection, there is renewal waiting to begin. In every fold that fails, there is the memory of how to fold again. And within you, as within all things, this quiet process continues a reminder that even the smallest flaws are part of the timeless art by which life learns to endure. 16. The beauty of molecular symmetry. In the vast and unseen world of molecules, there exists a kind of quiet perfection, a balance of shape, proportion, and pattern that seems almost artistic in its precision. Though life is built from chaos and chance, within that randomness, order arises again and again, weaving patterns of astonishing beauty. Proteins, those intricate architects of biology, are among the finest examples of this hidden symmetry. In their folds and curves, in the spirals of their helices and the lattices of their sheets, one can glimpse a natural geometry that rivals any work of human art. This symmetry is not merely aesthetic, it is the very language of function, the way nature ensures stability, efficiency, and grace. Through it, the living world achieves both form and harmony, a molecular architecture that sustains the rhythm of life itself. To observe a protein at the molecular level, even through the most powerful instruments, is to enter a world of astonishing design. What appears at first as a tangled mass of atoms soon reveals structure-repeating motifs, orderly layers, and patterns that recur across species and eras. The alpha helix, a delicate spiral that twists in perfect proportion, repeats every 3.6 amino acids, forming a structure as elegant as a coiled spring. Its turns are measured, its hydrogen bonds aligned with mathematical precision. Alongside it lies the beta sheet, where strands of amino acids stretch side by side, their hydrogen bonds forming a network as regular as woven fabric. These patterns are not accidental. They emerge naturally from the properties of the molecules themselves. In the watery environment of the cell, where thousands of forces act in concert, symmetry provides the calm center, the point where all competing energies find balance. The beauty of symmetry in proteins is not limited to their internal folds. Many exist as assemblies of multiple subunits, arranged in repeating patterns that create larger symmetrical structures. Hemoglobin, for example, consists of 4 subunits, 2 alpha and 2 beta chains, joined in a configuration of near-perfect balance. Each subunit is a mirror of the others, arranged around a central axis that allows them to cooperate in binding oxygen. When one subunit captures an oxygen molecule, the entire structure subtly shifts, making it easier for the remaining subunits to bind as well. This cooperative behavior, born of symmetry, demonstrates how elegance in form translates into efficiency in function. The same principle appears in countless other proteins, from enzymes that form hexagonal rings to viral capsids made of perfectly repeating units. The symmetry of these assemblies ensures strength and stability while minimizing the amount of genetic information needed to build them a lesson in nature's economy, where beauty and utility are one. The appeal of molecular symmetry extends beyond biology into the philosophy of existence itself. In every corner of nature, symmetry appears as a fundamental principle in the crystals of minerals, in the petals of flowers, in the spirals of galaxies. At the molecular level, it provides a bridge between chaos and order, showing how simplicity can give rise to complexity. The folding of a protein, for instance, is not directed by an external force, but emerges spontaneously from the interplay of atoms. Hydrogen bonds align, hydrophobic regions retreat from water, and electrostatic charges settle into harmony, producing a structure that seems deliberate but is born entirely of natural law. It is a reminder that beauty does not need intention. It arises naturally from balance, from the quiet insistence of order upon the world. In the living cell, this symmetry serves not only as decoration but as the foundation of stability. A symmetrical molecule distributes stress evenly, making it more resistant to disruption. The precise alignment of bonds allows for flexibility without fragility. This balance is essential, for life depends not on rigidity, but on adaptability. A protein must hold its shape well enough to perform its function, yet yield slightly to the movements of other molecules. The symmetry of its design allows both qualities to coexist. In enzymes, for instance, the symmetrical arrangement of active sites ensures that reactions occur with efficiency and consistency. In antibodies, the mirrored arms of the Y-shaped molecule allow it to bind to pathogens with precision and strength. Everywhere, symmetry acts as a quiet architect, building stability from motion and beauty from necessity. Even the smallest motifs within proteins exhibit this harmony. The arrangement of atoms around a central carbon, the geometry of peptide bonds, and the angles of side chains all conform to patterns of order. The chirality of amino acids, the fact that nearly all biological amino acids are of the left-handed form, adds a subtle asymmetry that allows the larger structures to twist and fold in consistent ways. Without this molecular handedness, proteins could not form the elegant helices and sheets that define them. Thus, even in asymmetry, there is purpose. Life's perfection does not come from flawless balance, but from the interplay of symmetry and its gentle disruption. It is in this tension that diversity arises from mirrored patterns slightly shifted, from structures that almost repeat but not quite. When scientists first began to visualize proteins through X-ray crystallography, they were astonished by the clarity of their patterns. Crystals of enzymes and structural proteins revealed repeating lattices that extended in all directions, their order more precise than anything the eye could imagine. Each atom had its place, each bond its purpose. The images, when translated into models, resembled miniature sculptures, spirals, spheres, and cages of breathtaking intricacy. In those silent structures, the boundaries between science and art seemed to dissolve. The beauty was not metaphorical but literal, an expression of the deep geometry of life, a glimpse of the symmetry that connects the molecular to the cosmic. Symmetry in proteins also mirrors the symmetry of behavior in nature. Just as molecules fold and assemble in predictable ways, so too do living systems organize themselves into patterns, from the hexagons of a honeycomb to the branching of blood vessels in trees. The same mathematical principles govern both the molecular and the macroscopic, linking the shape of a single enzyme to the growth of a coral reef. This continuity suggests that symmetry is not merely a feature of life, but one of its underlying laws. It allows for infinite variation built upon simple repeating foundations. The helix, for instance, appears again and again in DNA, in shells, in hurricanes, each manifestation a reflection of the same geometric truth. If one could drift through the molecular world as through a quiet gallery, the view would be wondrous. There would be the spiraling helices glowing softly in the fluid light, the lattices of beta sheets shimmering like woven silk, and the great assemblies of protein complexes turning slowly in symmetrical grace. Every form would seem to breathe with purpose. every repetition carrying meaning. This unseen realm is not static but alive, its symmetry dynamic and responsive, always adjusting to the needs of the cell, always maintaining balance amid change. It is a world where beauty is function and function is beauty, where design emerges not from thought but from the logic of nature itself. In human art and architecture, symmetry has always symbolized harmony, stability, and perfection. Temples, cathedrals, and mandalas all echo the patterns found in nature, reflecting an intuitive recognition of the same principle that governs molecules. When we find beauty in the curve of a shell or the proportions of a leaf, we are responding to the same geometry that shapes our own cells. The symmetry within us resonates with the symmetry around us, creating a sense of unity that transcends scale and form. Proteins, in their quiet complexity, embody this unity, the harmony of matter arranged for purpose, the grace of balance maintained within motion. As you rest and imagine the world within, picture these molecular symmetries unfolding endlessly. See the spirals, the mirrored shapes, the repeating patterns that sustain you. Within every heartbeat, every breath, these molecules continue their timeless dance of balance and form. They are unseen, yet they shape every sensation, every movement, every moment of being. The beauty of molecular symmetry lies not only in its precision but in its constancy, the assurance that, even in the smallest spaces, the universe creates order. In this hidden geometry, there is peace, a quiet truth that everything, from the stars above to the cells within, moves according to the same ancient harmony. 17. The connection between DNA and proteins. Every living cell is a world unto itself, humming with purpose and quiet design. Inside this microscopic universe, two great molecules define the essence of life: DNA and proteins. Between them lies a partnership as ancient as life itself, a relationship built on information and action, on language and expression. DNA holds the instructions, the blueprint of existence, while proteins carry out the work, building, repairing, and maintaining the structures that sustain being. The connection between them is intimate and unbroken, a continuous dialogue written in the soft rhythm of chemistry. To understand this connection is to glimpse the very mechanism by which life reads, interprets, and renews itself, transforming the stillness of information into the movement of living matter. DNA, or deoxyribonucleic acid, rests at the heart of every cell, coiled gently into spiraling strands that hold the instructions for all that the organism is and can be. Its structure, the double helix, is one of nature's most beautiful and efficient designs. Two long chains of nucleotides wind around each other like intertwined ribbons, bound together by hydrogen bonds between paired bases, adenine with thymine, cytosine with guanine. The sequence of these bases forms a code, not in words but in patterns of 4 letters, arranged in infinite combinations. Within this simple alphabet lies the full story of life: how a cell divides, how an eye perceives light, how a flower opens at dawn. Yet DNA itself does not act. It is a silent library, a vast storehouse of potential, waiting for something to give it voice. Proteins are that voice. They are the interpreters of the genetic script, the instruments that turn instruction into form. Every movement within a living organism, every heartbeat, every thought, every breath is made possible by proteins that were created according to the code inscribed in DNA. The process that connects the two, the flow of information from gene to protein, is known as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. This flow is unidirectional and precise, a chain of translation that converts information into existence itself. Within the nucleus of the cell, where the DNA resides, enzymes called RNA polymerases move along the DNA strand, reading the sequence of bases like a careful scribe. As they progress, they build a complementary strand of messenger RNA, mRNA, substituting uracil for thymine, but preserving the genetic message. The mRNA acts as a copy, a portable version of the instructions, small enough to leave the nucleus and enter the bustling cytoplasm. There, it becomes the template for the next stage. Translation, the act of building a protein. Translation occurs on the ribosome, a molecular machine made of both RNA and protein. The ribosome reads the mRNA 3 bases at a time, each group of 3 forming a codon, and each codon corresponding to one specific amino acid. It is a language written in triplets, with 64 possible codons forming the vocabulary of life. Some codons specify amino acids, while others mark the start or end of a sequence. Transfer, RNA, tRNA. Molecules act as interpreters, carrying individual amino acids to the ribosome and matching their anticodons with the codons on the mRNA. One by one, the amino acids join, forming a growing chain that folds into the unique shape of a protein. This process, so intricate yet so natural, is the bridge between the genetic code and the living body. It transforms abstract information into tangible form. Every cell, whether in a leaf, a muscle, or a nerve, carries the same DNA. Yet it produces different proteins according to its needs. A muscle cell expresses the genes for actin and myosin, giving it the ability to contract. A nerve cell produces proteins that transmit electrical impulses. A skin cell builds keratin to provide protection. The DNA remains the same, only the expression changes. This selective reading of the genome is what allows a single fertilized egg to grow into the vast diversity of tissues that make up a living being. The relationship between DNA and proteins is not merely mechanical, it is deeply cyclical. While DNA gives rise to proteins, proteins, in turn, protect and regulate DNA. Enzymes called polymerases and helicases, themselves proteins, are responsible for copying and repairing DNA during cell division. Other proteins package DNA into chromatin, folding it neatly so that it can fit within the tiny space of the nucleus. Regulatory proteins decide which genes are active at any moment, opening or closing access to specific regions of DNA. In this way, DNA and proteins form a feedback loop, one creating the other maintaining the first and endless cycle of interdependence that keeps life balanced and self-sustaining. It is extraordinary to consider how this molecular conversation has persisted through billions of years. From the first primitive cells in ancient oceans to the intricate forms of life that fill the world today, the same fundamental connection has remained unchanged. The code written in DNA is universal. A gene from a bacterium can be read and expressed by a human cell, and it will produce the same protein. This universality speaks of a shared ancestry, a single origin from which all life descends. Across time and species, the connection between DNA and proteins has bound every organism to the same molecular heritage. It is as though all living things are variations of one great theme, their differences shaped not by the alphabet itself, but by how it is arranged and read. The elegance of this system lies not only in its precision, but in its adaptability. Mutations, small changes in the DNA sequence, occur naturally over time. Most are neutral or harmful, but a few create proteins with new properties, new shapes, or new functions. Through natural selection, those changes that benefit the organism are preserved, passed down through generations, and incorporated into the evolving language of life. In this way, DNA and proteins together create the possibility of change while maintaining stability. The code endures, but the expression evolves, allowing life to explore new forms without losing its foundation. This connection also mirrors a profound philosophical truth that information and matter are two aspects of the same reality. DNA holds the instructions. Proteins give those instructions form. One cannot exist meaningfully without the other. The gene without the protein is an unread book, silent and inert. The protein without the gene has no source, no continuity. Together they form a self-renewing system, information creating form, and form preserving information. Within this partnership lies the mystery of life itself, the way consciousness, growth, and renewal arise from the steady pulse of molecular communication. If one could watch this process unfold within a cell, it would appear like a living script being written and rewritten in real time. Strands of DNA unwind gently, exposing their sequences. RNA polymerases glide along them like moving pens, tracing lines of code. The mRNA emerges, carrying its message through the fluid of the cytoplasm. Ribosomes gather around it, reading its triplets and assembling chains of amino acids with rhythmic precision. The newly formed protein folds upon itself, adopting a structure as intricate as lace. Around it, other molecules move in harmony, enzymes refining, chaperones guiding, membranes vibrating softly. The entire scene would feel like music rendered in motion, the silent symphony of life composing itself again and again, and within you, this symphony never stops. Even as you rest, cells continue to read their DNA, produce their proteins, and replace those that have aged or been damaged. The connection between DNA and proteins is not a single event, but a constant flow, a river of creation that never ceases. It is what keeps you alive, what allows your heart to beat, your lungs to fill with air, your mind to dream. It is the thread that ties your present moment to the deep past, linking you to every living being that has ever existed. In the quiet of reflection, one might think of DNA as memory and protein as expression, two halves of the same whole. Memory gives life continuity. Expression gives it movement. Without one, there is no identity. Without the other, no action. Together, they embody the unity of being the silent intelligence that pervades every cell, the calm rhythm that sustains all existence. The connection between DNA and proteins is, in truth, the connection between stillness and motion, between idea and reality, between what is imagined and what is made real. As you drift deeper into calm, imagine this partnership continuing within you, steady and eternal. The DNA rests, silent and perfect, holding the record of your life. The proteins move and act, carrying out the dance that keeps you whole. Between them flows an unending conversation, older than the stars, whispered in the language of molecules, the language of life itself. 18. Proteins in motion, molecular machines. Deep within the living cell, beyond the reach of sight, there is a world that never stands still. Molecules drift, collide, and interact in a quiet rhythm that has been unfolding since the beginning of life. Among them are the proteins, intricate structures that not only exist in graceful symmetry, but also move, turn, and work with purpose. Some are still and structural, providing the strength and form that give life stability. Others, however, are in constant motion, performing tasks that seem almost mechanical in their precision. These are the molecular machines of life, proteins that move, rotate, and transport, their motions measured and deliberate, yet born entirely from the simple rules of chemistry. They are the silent engines of biology, transforming energy into action, ensuring that every process within the cell continues in balance and harmony. To think of a protein as a machine may seem strange, for we are used to imagining machines as human inventions, made of metal and wire, powered by fuel or electricity. Yet in truth, the cell's molecular machines predate all others by billions of years. They operate not through conscious design, but through natural order, harnessing the energy of chemical bonds to perform work with efficiency that surpasses anything built by human hands. Each one is an assembly of atoms arranged so precisely that it can move in specific ways, flexing, spinning, or stepping, all without sound, all within the gentle flow of the cell's fluid interior. At the heart of this molecular world, one might find motor proteins, remarkable molecules capable of producing movement on a scale so small that their steps are measured in billionths of a meter. Among them are myosin, kinesin, and dynein, each a marvel of natural engineering. Myosin, found in muscle cells, converts chemical energy from adenosine triphosphate, ATP, into mechanical force. It binds to filaments of actin and pulls against them, causing the contraction of muscle fibers. When billions of these interactions occur together, the result is movement, the lifting of an arm, the beat of a heart, the blink of an eye. The same process that powers every heartbeat and breath arises from countless microscopic motions, each one repeating its cycle with unwavering precision. Kinesin and dynein work in a different way but with equal elegance. They are transporters, carrying materials across the vast interior of the cell as though walking along invisible highways. Kinesin moves toward the outer regions of the cell, while dynein travels inward toward the nucleus. Each has two globular heads that alternately bind and release from microtubules, moving forward step by step, much like feet walking across a path. With each step, a molecule of ATP is consumed, its energy released and converted into motion. Watching kinesin under a microscope, as scientists have managed to do with advanced imaging, is to see life's machinery revealed. A single molecule striding patiently along a filament, carrying its tiny cargo to where it is needed. This motion is astonishingly efficient. A kinesin molecule can take hundreds of steps without error, guided solely by the shape of its structure and the timing of chemical reactions. It does not think, yet it behaves as though it knows where to go. It does not see, yet it navigates the crowded, shifting environment of the cell without colliding with other structures. It performs its function flawlessly, driven by the simple yet profound rule that energy flows toward balance. In every movement, in every step, almost alive. Other molecular machines perform different kinds of motion. The ATP synthase enzyme, for instance, is one of the most remarkable examples of rotary motion in biology. Found in the membranes of mitochondria, the powerhouses of the cell, this enzyme generates ATP. Its structure resembles a turbine, with a rotating shaft driven by the flow of protons across the mitochondrial membrane. As the protons move through, they turn the central rotor, which in turn drives the formation of ATP from its components, adenosine diphosphate and inorganic phosphate. Each rotation produces 3 molecules of ATP, the fuel that powers nearly every cellular process. This rotary engine, smaller than a virus, spins at speeds of up to several hundred revolutions per second. Within it, we find a glimpse of perfection, a machine so efficient that it wastes almost no energy, endlessly producing the molecules that sustain life. There are other forms of motion as well: twisting, unwinding, assembling, and disassembling. The helicases, for example, are proteins that unwind DNA strands during replication, moving along the double helix like careful readers unrolling a scroll. Each step requires energy, drawn from ATP, and each ensures that the genetic code can be copied accurately for the next generation of cells. Similarly, polymerases move steadily along DNA or RNA templates, adding nucleotides one by one, constructing the molecules of heredity with unerring precision. These molecular workers never tire, never stop to rest. They move at their assigned pace, guided by shape and chemistry, not by thought or will. Even within the membranes that encase the cell, motion never ceases. Ion pumps and channels open and close in rhythm, transporting charged particles like sodium, potassium, and calcium across the lipid boundary. These movements generate electrical gradients that allow neurons to fire and muscles to contract. Sodium-potassium pump, one of the most important of these systems, maintains the delicate balance of ions that keeps cells alive. It works like a heartbeat at the molecular level, pumping ions in and out with every cycle, using ATP as its fuel. Without this constant motion, the electrical signals that govern thought, sensation, and movement would vanish within moments. The harmony of these molecular machines is astounding. Each type of motion, linear, rotational, oscillating, contributes to the greater function of the cell. Some work alone, others in vast assemblies, coordinating their efforts like the instruments of an orchestra. There are ribosomes, which build proteins, spliceosomes, which edit RNA, proteasomes, which break down unwanted proteins, and countless others, each performing its task in perfect time. Together, they transform the cell into a living factory, a place where energy becomes structure, where chemistry becomes motion, and where every movement serves the quiet continuity of life. Though we cannot see them with our eyes, the activity of these molecular machines defines our existence. Every movement of your body, every heartbeat, every breath, every thought is the sum of countless microscopic motions happening in unison. Within your muscles, myosin molecules pull on actin filaments, shortening fibers to create motion. In your brain, ion channels open and close, allowing electrical impulses to travel. In your mitochondria, ATP synthase spins tirelessly, supplying the energy for it all. Life, in its essence, is motion, not chaotic motion, but motion guided by order, by balance, by the quiet laws that govern the universe at every scale. If you could imagine yourself shrinking down to the scale of atoms, floating within the fluid interior of a cell, you would see a landscape alive with movement. Filaments stretch and contract, vesicles glide along microtubules, enzymes twist and turn in perfect rhythm. It would not be a scene of chaos, but of harmony, each motion precise, each interaction meaningful. The cell would feel less like a machine and more like a living poem, written in motion rather than words, where every molecule plays its part in the unfolding of life's endless verse. As you rest, this motion continues within you, steady and unbroken. Myosin molecules pull in your heart, ion channels open in your nerves, and countless enzymes carry out their silent tasks. None of them pause. None of them falter. They have been performing this same work since before you were born, and they will continue until your last breath. Their movement is the pulse beneath all things, the quiet machinery that sustains life, unseen, yet constant. In the stillness of reflection, there is something profoundly beautiful about this ceaseless motion. It reminds us that even when we are still, we are alive with activity, our bodies composed of countless tiny worlds in motion. The proteins within us are not static matter, but living, energy, transforming, moving, and renewing with every moment. They are the dancers in the great choreography of existence, their steps measured not in seconds, but in molecules. Within their silent movement lies the heart of life, a song without sound, a dance without end, unfolding softly in the depths of every cell. 19. The harmony of biochemical systems. Within the vast and intricate interior of every living cell, there exists a profound order, a harmony so complete that countless reactions occur every second without conflict or confusion. It is a quiet symphony of balance and precision, a choreography of molecules that never falters. Each component knows its place. Each interaction flows into the next, and together they sustain the miracle of life. This harmony of biochemical systems is what allows a single cell to breathe, to repair, to sense its environment, and to respond. It is the music of chemistry transformed into the poetry of existence, where countless molecular voices blend into a single, living rhythm. To imagine this harmony, one must picture a cell as a city not of stone or light, but of motion and exchange. Within its boundaries, proteins, lipids, sugars, and nucleic acids work as citizens of a bustling metropolis. Each performs its duty with precision. Some carry energy, others build structures, some send signals, while others clean and renew. Yet there is no chaos here, no traffic or noise. Every process occurs in coordination with others, guided by feedback loops, molecular signals, and gradients of energy. The cell is not a random assembly, but an ecosystem, where every reaction supports another, where balance is maintained not by control, but by connection. At the heart of this harmony lies metabolism, the sum of all the chemical reactions that occur within a living organism. Metabolism is the constant exchange between energy and matter, between building up and breaking down. It has two complementary sides: anabolism, which constructs and stores, and catabolism, which breaks down and releases. Together, they form a cycle of creation and renewal. Through catabolism, nutrients from food are transformed into simpler molecules, releasing energy stored in chemical bonds. That energy, captured in molecules like adenosine triphosphate, ATP, fuels anabolism, the synthesis of new proteins, lipids, and nucleic acids. The balance between these two processes ensures that the body neither starves nor overflows with excess, that it moves steadily through the flux of time with quiet efficiency. Energy itself flows through this system like music through air, unseen but essential. It begins with the sun, whose light is captured by plants through photosynthesis. In the chloroplasts of their cells, light energy transforms carbon dioxide and water into glucose and oxygen. This glucose becomes the fuel for almost all living organisms, passing from plants to animals through the food chain. Within our own cells, glucose enters pathways like glycolysis and the citric acid cycle, where it is broken down step by step. Each step releases energy in small, controlled amounts, preventing waste and allowing life to persist. The energy released forms ATP, the molecule of exchange that carries power to every corner of the cell. Through ATP, chemical energy becomes motion, thought, and warmth. The cell, like a perfect instrument, never lets this energy rest idle. Every molecule is used, every atom accounted for. This coordination of energy and matter depends on enzymes, the catalysts of life. Each enzyme has its role, accelerating one specific reaction and ensuring it occurs at the right speed, in the right place, under the right conditions. Without enzymes, the chemistry of life would move too slowly to sustain existence. With them, reactions occur millions of times faster, yet still under control. Some enzymes work in chains, passing products from one to the next, like dancers in sequence. Others are regulated by signals that tell them when to begin or stop, preventing waste and imbalance. In every pathway, enzymes maintain the harmony of metabolism, ensuring that energy flows like a continuous melody, never too loud, never too soft. Communication between these systems is equally important. The cell must constantly sense its environment, the availability of nutrients, the presence of toxins, the demand for energy, and adjust its activity accordingly. Signaling molecules, such as hormones and secondary messengers, act as the language of coordination. They move through the cytoplasm or across membranes, carrying information from one region to another. When glucose levels rise, insulin signals cells to absorb it. When energy is low, other molecules signal the need for more ATP production. This constant communication prevents extremes, guiding the system gently back toward equilibrium whenever it drifts too far in one direction. The result is a living stability, an equilibrium that is never still, but always in balance. Yet harmony in biochemistry is not merely about molecules acting in isolation. It is about networks. Every pathway, every reaction is connected to others. The product of one becomes the raw material of the next, and together they form cycles of renewal. The citric acid cycle, for example, links energy metabolism to the synthesis of amino acids and lipids. The urea cycle connects protein metabolism to nitrogen balance. These cycles interlock like gears in an invisible engine, each turning in time with the others. If one slows or falters, the rest adjust, shifting their pace to compensate. This interconnectedness gives life resilience, allowing it to adapt to changing environments and recover from disruption. The harmony of biochemical systems extends beyond individual cells, uniting entire organisms. In multicellular beings like humans, each tissue has its own specialty, its own rhythm, yet all remain connected through blood, nerves, and hormones. The liver balances glucose production with storage. The muscles consume energy when they move. The brain regulates appetite, stress, and temperature, ensuring that every part of the body receives what it needs. This orchestration is so seamless that we rarely notice it. Even as we sleep, countless systems continue to converse: the lungs, exchanging gases, the heart maintaining rhythm, the kidneys filtering and restoring balance. Life is not a series of isolated reactions, but a single, continuous conversation written in the language of chemistry. There is an elegance to this balance that mirrors the patterns of the natural world. Just as an ecosystem finds harmony between predator and prey, sunlight and shadow, so too does the body find equilibrium between energy and rest, between growth and decay. The same principles that shape forests and oceans echo within the human cell. Waste becomes resource, motion becomes rest, and every end feeds a new beginning. The law of conservation, the flow of balance, and the return to equilibrium, all are as true for molecules as they are for galaxies. Even the smallest deviation can ripple through this system, yet the body's design ensures that balance is restored. When blood sugar rises, insulin responds. When oxygen falls, breathing deepens. When heat increases, sweat cools. These are not conscious decisions, but automatic harmonies, the body's way of preserving life without thought. The same mechanisms exist within each cell. Sensors that detect imbalance and proteins that respond. Every reaction is tuned, every process reversible if needed. It is an endless symphony of correction, one that plays softly beneath the surface of awareness. If one were to visualize this molecular harmony, it would resemble a landscape in motion, streams of molecules flowing like rivers, enzymes acting as gates that open and close in rhythm, ATP molecules flickering like sparks of light. Within this landscape, everything moves toward balance, each reaction finding its place in the greater whole. It is neither mechanical nor random, but organic, a self-sustaining rhythm that has continued unbroken since the dawn of life. As you rest and breathe, this harmony continues within you. Every cell plays its part in the music of existence, transforming food into energy, energy into action, and action into renewal. Even now, as your body lies still, your heart beats in quiet rhythm, your lungs exchange oxygen for carbon dioxide, and your cells orchestrate thousands of reactions with silent precision. This is the peace of life, not the absence of motion, but motion so balanced that it feels still. In the end, the harmony of biochemical systems is not something apart from us. It is what we are. Every sensation, every thought, every breath arises from this balance. Within each heartbeat, within each molecule, there is order, purpose, and connection. The body is not a machine, but a symphony, a living composition written in atoms, conducted by the forces of nature, and performed endlessly in the quiet theater of existence. It is the most ancient song, one that began in the first cell and has never ceased to play. Its melody is the sound of life itself, delicate, perpetual, and perfectly in tune. 20. A gentle closing reflection. As the journey through the quiet science of amino acids and proteins draws to its close, we find ourselves returning to where it all began, to the calm recognition that life, in its essence, is both simple and infinitely complex. Every breath, every heartbeat, every moment of awareness is built upon the work of these silent molecules, folding, moving, and interacting in patterns too small to see, yet grand enough to encompass the entirety of existence. In their microscopic dance lies the story of life itself, written not in words or sounds, but in the language of chemistry and balance. Proteins, the humble builders and caretakers of all living things, continue their work without pause, their harmony unbroken by time. They are the invisible caretakers of our being, the quiet artisans shaping each heartbeat, thought, and breath. To reflect on their beauty is to reflect on life's most intimate mystery, the transformation of stillness into motion, of structure into feeling, of matter into consciousness. A single amino acid, so small it cannot be seen, links with others in precise sequence, guided by the patterns held in DNA. From this simple chain arises a folded structure, and from that structure emerges function, a muscle that contracts, an enzyme that catalyzes, a receptor that senses. Within every fold, every interaction, there is purpose. It is as though nature itself speaks through these molecules, whispering that meaning does not need to be sought beyond the material world, for it already resides within it. The human body is not a separate entity from this quiet molecular world. Its expression, an unfolding of chemical harmony into physical and emotional form. The same proteins that hold together the structure of a leaf, that let a fish glide through water or a bird take flight, are reflected within us. In that shared chemistry lies kinship, the deep connection between all living things. Whether one looks at the structure of hemoglobin in the blood or chlorophyll in a plant's leaf, the resemblance is clear. Both are complex. Both carry the mark of the same elemental forces. And both remind us that life, in all its forms, is united by design. The differences that separate species are only variations on an ancient theme, small changes in sequence, small shifts in shape, each one opening a new path in the endless exploration of being. The harmony of proteins, the balance of reactions, the flow of energy, all of it happens without conscious command. The body does not think about maintaining its temperature, repairing its tissues, or generating energy. It simply does. Life continues as it always has, through the quiet intelligence of chemistry. This intelligence is not thought or will as we understand it, but a deeper order, one that arises from the properties of atoms and the forces between them. It is as if the universe itself, in its timeless stillness, found a way to move through us, expressing its equilibrium in the rhythm of our cells. There is something profoundly comforting in this realization. It reminds us that we are not separate from the processes that sustain us. Even in stillness, we are part of movement. Even in rest, we are part of creation. The same laws that shape galaxies shape the folding of proteins. The same patterns that guide the flow of rivers guide the flow of blood. There is no division between the vast and the minute, only scale. And within that continuity, we find peace, the understanding that everything that exists, from the smallest amino acid to the greatest star, story. As we reflect on this story, it becomes clear that life's quiet mechanisms mirror the patterns of nature around us. The folding of proteins is like the curling of a fern frond, the coiling of a seashell, or the spiral of a galaxy. Each follows the same principles of symmetry, energy, and balance. When a protein folds into its proper shape, it finds the point of least resistance, the most stable arrangement possible, where all its forces align in harmony. In much the same way, nature itself always seeks balance. The tides find equilibrium between moon and sea, the seasons turn in measured cycles, and the living world sustains itself through endless renewal. Every fold in a molecule, every interaction within a cell, reflects this universal search for balance. Even imperfection, as we have seen, plays a role in this great design. Proteins misfold, reactions falter, and yet the system endures. It adapts, corrects, and learns. Within these errors lie the seeds of change, the potential for evolution, the gentle hand of time shaping new possibilities. It survives not by avoiding change, but by flowing with it. This, too, is a kind of symmetry, the balance between creation and decay, between perfection and imperfection. When one contemplates the unity of these processes, a kind of calm reverence emerges. Within it, molecules meet, transform, and part ways, each following the laws of thermodynamics, yet collectively creating something greater than the sum of their parts. This is the miracle of emergence, how order arises from simplicity, how awareness emerges from chemistry. The mind that contemplates this miracle is itself a product of it, a pattern of proteins and neurons, shaped by the same laws it seeks to understand. In this way, the act of reflection becomes circular, life reflecting upon life, consciousness aware of the molecules that made it possible to lie still and breathe deeply is to participate in this harmony. With every breath, oxygen enters the lungs, binds to hemoglobin, and travels to every cell. There, it fuels the reactions that generate energy, allowing proteins to move, repair, and renew. Each heartbeat carries this rhythm through the body, a reminder that motion and rest coexist in perfect balance. Even as the mind drifts into stillness, the body remains alive with motion, enzymes folding and unfolding, ions flowing across membranes, proteins assembling and dissolving. This quiet symphony never ends. It began long before birth and will continue, in different forms, long after. In the gentleness of reflection, one might realize that this molecular harmony extends beyond the boundaries of biology. The patterns of proteins, the flow of biochemical reactions, and the structure of DNA are all part of a wider order, the same order that shapes water into waves and stars into galaxies. Life, then, is not an accident, but a continuation of the universe's desire to organize itself, to create patterns from chaos. Through proteins and their elegant forms, the universe has found a way to look upon itself, to feel, to think, to wonder. Each of us, built from these same molecules, carries within our cells the quiet wisdom of the cosmos. And so, as this gentle exploration comes to an end, we return to the stillness from which it began. The science of amino acids and proteins is not just a study of molecules, but a meditation on connection. It teaches us that life is not separate from matter but born from it, that beauty is not added to the world but inherent within it. The same forces that guide the folding of a protein guide the unfolding of our lives, subtle, balanced. As you breathe and rest, imagine the countless proteins within you, still at work, shaping, repairing, and renewing. They do not sleep. They do not tire. They move in the rhythm of life itself, their silent song echoing through every cell. You are their creation, their masterpiece, their harmony made visible. In the depths of your being, chemistry becomes consciousness, structure becomes self, and motion becomes meaning. The science of life is, at its heart, a story of peace, a reminder that even in complexity, there is calm. Even in motion, there is rest. And even in the smallest particle, there is the reflection of eternity.